So 2 minutes.
Okay.
Akila, start at sharp 9. Akila and Pooja, start at sharp 9. Can you hear me? Can you hear me, Akila? Akila, can you hear me?
Yes, sir.
Okay. Start at sharp 9.
Okay. Now I'm going to
A very good morning, ladies and gentlemen. This is Akhila, and I welcome you all to the Day 2 of International Symposium on Bioinformatics for Disease Therapeutics. I know you all bagged a lot much of knowledge and data yesterday, and today we are going ahead with Technical Session 3 and Technical Session 4. Starting with Technical Session 3, I would like to request Dr. Nazneen Bobby, Associate Professor, Department of Biotechnology, Vignan's Foundation for Science, Technology and Research, to introduce the keynote speaker. Ma'am, please.
Am I audible, sir?
Yes, madam.
Good morning, one and all. Welcome to the second day of this International Symposium on Bioinformatics. I'm very much delighted to give the brief bio data of Professor Gajendra Raghava. Professor Raghava received his MTech from IIT Delhi in 1986 and PhD from Punjab University in Tech Chandigarh in 1996. He worked as postdoctoral fellow at Oxford University, UK, bioinformatics specialist at UMS, USA, and visiting professor at postdoc South Korea in the year 2004. He worked around 30 years at different scientist positions in the field of bioinformatics at CSR Institute of Microbiology Technology, Chandigarh. Presently, he is working as professor at IIIT Delhi in the field of computational biology. He has published more than 250 research papers in reputed journals. He got Thomson Reuters Research Excellence India Research Front Awards in 2009. Because of his two papers, he ranked in the category of highly cited papers in the world. In 2014, he has been listed in the world's most influential scientific minds in the field of computer science. His group developed more than 250 in silico products, which is highest contribution by a single group in the world. He is a strong supporter of open source software, web servers. All services developed at his group are free for academic use. These web-based services are heavily used worldwide, more than 150,000 hits per day. He got numerous awards and recognitions. Few have been listed below. So I'm going to read one of Shanti Swarup Bhatnagar Award in Biological Science in the year 2008, National Bioscience Award for Career Development in the year 2006, J.C. Bose National Fellowship from the year 2010 to 20 by DST, Fellow of National Academy of Science, Fellow of Indian Academy of Science, and he is also the Fellow of Indian National Science Academy. With this, I request Professor Gajendra Raghava to deliver his talk. Sir, please.
Sir, please unmute yourself.
Hi.
Hi, sir. Sir, I had enabled the screen sharing. Please share your PPT and present.
Just hold.
Okay, sir.
It is showing disabled sharing actually. You have to allow me to share.
Sir, please share, sir. Already had enabled you.
Okay. Is it shared?
Yeah, we can see, sir. We can see it.
Okay.
Anil, tell professor to have this full screen display.
Yes, sir.
So tell me when I should to start.
So you can start now, sir.
All right. So today talk is about the computer-aided immunotherapy to fight against emerging pathogens. Right. So most of the people in bioinformatics, they mainly focused on the drug discovery, but our group, one of the major focus is on immunotherapy or vaccines. So I will go through each and every step. Let's say just come straight to understand the immune system. Why we got the disease? There are two type of disease. One is through the attack by the pathogens. Maybe they are called microbes basically. Bacteria, fungus, parasites, virus. These are some of the examples. Okay. Or the disease may be due to the disorder of our own own body system basically. Okay. So I'm talking about disease caused by pathogens. Maybe it's due to bacteria, virus, this sort of thing. Before I talk further or explain the concept further, it is important to understand the defense system actually, because without doing, without understanding biology or immune system, you cannot develop the tools for bioinformatics. That's the most important part. So let's say when any enemy or disease causing agent attack a human, so what will happen? There will be two type of defense. First is non-specific defense, or we call innate immunity. Innate immunity will come into the picture, and they will release certain chemicals, certain toxic materials, so that they will kill the disease causing agent, bacteria, fungus, whatever, pathogen basically. And these pathogens are recognized by the receptors actually, that is called pattern recognition receptor, PRRP, and main class of the PRRP is toll-like receptors basically. Okay. So they recognize pathogen associated molecular patterns actually, because each this pathogen, bacteria, fungus, they have certain type of pattern on the surface actually, and this is recognized by the innate immune system, and they give the signal to the body, look, there is an enemy, and same time they release certain chemicals to provide the first line of defense. So this is the one part of the system, innate immunity. There is a specific defense. Still there is some virus or bacteria which crosses this innate immunity actually, which bypass actually. So then there is a next system that is called specific defense system or adaptive immunity. In case of the adaptive immunity, then there is a two part, humoral immunity and cellular immunity. In humoral immunity, there is a B cell basically, which release the antibodies which can directly neutralize the pathogen. There is another category that is called cell-mediated immunity, where they have the T helper cell and cytotoxic T cells basically. These are the two categories, and these cytotoxic T cells, they release certain chemicals and they kill the infected cell, where the T helper cell activate both arm, means B cell as well as cytotoxic T cells. So let's say for as a bioinformatics, what is challenge? Challenge is initially the vaccines are basically based on the whole pathogen. They take the whole pathogen, they kill it or annihilate it and give to the body. So what will happen? Then there will be immune system will thought that enemy is there and then defense will be prepared. During the defense preparation, they will generate the memory cells. They may be called the they called there is echo. Hello. Yeah. Now it's so there is a hello. So there is a memory piece and memory piece. Something is going wrong actually.
It's giving resound, sir.
Hello. There is an echo actually.
Yes, sir. Maybe phone is near to you. You just check out, sir.
You make everyone mute, sir.
Hmm?
You make everyone mute except Professor Raghav.
All right. Hello. Yes. Now it's okay. Oh. Okay. There is something. Hello.
Professor Raghava, sir?
Yes.
Yeah, you have logged into two computers, I think. So kindly switch off one of the computers. So
Just hold on. I will.
yeah, this resound is because you have logged into two computers, sir.
Just hold on.
Yeah. So kindly switch off one of the computers.
Hello.
Yeah, sir. We hear you.
Sir, please unmute yourself and
Yeah. Hello. I don't know why there is too much echo. Hello. Now it's no, some of your systems are not mute actually. I think system administration can do the mutation, mute the others actually.
except you all are on mute only, sir.
No, this is number is 18 earlier. Now it's okay. I don't know why. Hello. Don't know what is going on.
So if you don't mind, could you rejoin again?
Sorry to interrupt, but Jyotika Bhatti's mic is on. Can you please mute Jyotika?
Okay, I will do one thing. I will leave it and then I will join it again. Actually, maybe it will help.
Okay, sir.
Hello.
Yes, sir, we can hear you, sir.
Yes. Okay. I will share the screen. Now my presentation is visible.
Yes, sir, we can see it.
Okay. Okay. Fine. So I was talking about the B cell and T cell memory generations. Now I will go to the next part. Okay. So first we have to understand that how the antigen is processed, how a pathogen is processed basically. Let's say if bacteria or virus. Okay, so if any pathogen goes inside the cell actually, what will happen actually? Let's say this is the example that pathogen is here, which is a disease causing pathogen, and what will happen? Because these pathogens, these are the living organisms, they also release certain type of proteins basically. These are called the secretory proteins. Okay, these secretory proteins are not the same as our human proteins. So we have the proteasome system in our body. This proteasome recognizes foreign proteins, and once they recognize it, they chop it into the pieces actually. PCs in the means term of, let's say, small peptides, 9 amino acid peptide, 10 amino acid peptide, whatever is there, they chop it depending on the enzyme cleavage site basically. Now question is basically how to predict that what type of fragment will be generated from a pathogen protein actually. So there are a number of softwares have been developed in the past. Our group also developed a software called p-cleavage. This is our group software where we predict whether this protein will be cut or not. If it is cut, then from where it will cut, means what fragment will be generated from a protein. So this is the one part, you get the peptide corresponding to a protein. The next part is there is a ER system, endoplasmic rectum is there, it has to go there actually. Okay, then there is a TAP proteins are there which work as a receptor. So they only the peptide can go inside the ER which can bind to the TAP actually. So we need to predict, we need to develop a method which can predict whether a given peptide will bind to the TAP or not. So that's the second condition. So number of groups have developed methods. Our group developed the method called TAP-PRED, and this TAP-PRED allow to predict whether a peptide will go inside the ER system or not. Once the PR is in this peptide, selected peptides is inside the ER, there is a MSC class 1 molecule, or we call HLA1. HLA1. There are different alleles of HLA1. Okay. Then some of the peptide will bind to HLA, not all. The question is which peptide will bind to the HLA. That's a challenge. So we developed the method to predict which peptide will bind to which HLA allele actually. So that method is called ProPred1 from our group, and there are a number of other groups who are also involved in this business. They also develop the tools actually. These tools are available. Once a peptide of a pathogen bind, then it will take the HLA molecule, will take the bound peptide to cell surface. It means ideally the pathogen antigenic region is ultimately taken by the HLA to the cell surface. Here is a CD8 cells are there, or called cytotoxic T cells. These are cells which are monitoring the system if there is any infection in any cell. Once they find out that one MSC molecule or HLA molecule is having the bounded peptide, then they will bound to it, T cell receptor will bind to it and will recognize that cell is infected. So then what will be the what will happen? Then CD8 cell will release certain type of cytokines, and these cytokines will kill the infected cell. So that's a mechanism for killing the infected cells from the body, just to clear the infected cells from the body. So here we have a method for predicting cytotoxic T cell for MSC binders. Okay. So here is requirement of immunoinformatics. For each and every step, we need to develop the method to predict the right candidate. Because if you do the same thing with the experimental techniques, it will take a long time actually. Okay, so practically it's not possible. So with the help of the computer, you can find out which peptide will activate our CD8 cells. So there is two actions here. CD8 cell will release certain cytokines and will kill the infected cell, as well as the cell which recognize this part will generate the memory B cells, memory T cells, so that in future, if there is an infection, then sufficient number of CD8 cells which can recognize this pathogen is there in the system. That's the basically concept of the vaccine. So whole life you have the large number of T cells which recognize a particular pathogen.
Okay. This is another concept. When your pathogen is outside the cell, actually. In this situation, pathogen is not inside. Here is a pathogen. Okay. So they are releasing a certain type of proteins only, actually. They are outside the cell and releasing a certain type of proteins, basically. These proteins will be engulfed by your antigen presenting cells. These antigen presenting cells will take this engulfed protein or antigen into the lysosome, actually. Lysosome is full of enzymes, basically. They will chop it into the different pieces. So there will be protein will be cut into the different peptides, actually. Once they are cut into peptides, then they will go bind to MSC class 2 molecule. Please remember this thing. Earlier I was talking about MSC class 1 or HLA1. Here I am talking about the MSC class 2 or HLA2. Okay. HLA and MSC class 2 is same thing. Only thing, case of the human, we call the human leukocyte antigen, actually. That's why we call HLA. Generally the word is MSC, major histolytic complex. Okay. Then it will be again presented on cell surface. This is a MSC molecule, this part is a MSC molecule, and this is a bounded peptide here. Okay, so this will be recognized by CD8 cells. Please remember, earlier it was CD8 cell. Here it is CD4 cell. So the concept is, if peptide is presented by MSC class 2 molecule, then CD8 cell will recognize it. If it is presented by class 1 molecule or HLA1 molecule, then it will be recognized by CD8 cell. That's a major fundamental difference. Now, question is, can we predict which peptide will bind to the MSC? So we have the software MSC, this ProPred, which will help you. There is other softwares also from our group, MSC, 2Pred and HLA Pred. They help to predict whether a certain peptide will bind to the particular MSC class 2 allele or not. Now, CD8 cell, or this is called the T helper cells, actually. They also have their branch TH1, TH2, and they release certain type of cytokines, actually. Some of them are basically interferon gamma, interleukin 4, like this, actually. Okay. So these are the methods where our methods simply predict this IFN epitope, predict whether it will release or induce interferon gamma or not, actually. These are particular type of cytokines, actually. So it will not only tell that it's a MSC binder, it will also tell whether it will release the interferon gamma or IL-4, actually. Because sometimes you need the specific type of cytokines in order to handle the particular type of disease. Okay, so there is another software called ProPred, actually, help to find out the MSC class 2 binders. And beauty of this software is that it helps you to identify the region where the most of the MSC class 2 alleles will bind. That's important to design a vaccine which can work for the wide range of population, actually, rather than for the specific class of population, actually. So that is called promiscuous MSC binders or epitopes. So I'm not going in the detail. These are the different methods. Similarly, there is a methods we have developed for predicting the B cell epitope. It may be conformation B cell epitope. It may be linear B cell epitope. So if you are interested, you can use them. So it started long time back, 20 years back. We realized that there is no database for the B cell epitopes, actually. So we collected and compiled this information, and then we created a database. This is important part. That's what we changed the trend, actually. Most of the cases, if you see the history of Indian science, actually, that most of the time database is created by the big countries like US, UK, and other countries. We are mostly users of them, actually. That gives a lot of disadvantage to us, actually. But what we did, actually, we created the databases, which are the primary databases. So anybody who want to work in the field, they can use this database. That's the important part, actually. So we have the source of the data. and we evaluate the first instance the all existing B-cell approach prediction methods, and then we realize the existing methods at the time in 2003 that most of the methods they have the poor performance, because 50% you can get by chance and they are getting maximum 57%. Then we combine all these physiochemical properties and then we reach up to 58.7%. That was the maximum achieved by any method actually. Okay. There is a first method we developed using the machine learning technique for B-cell approach prediction actually. Before that, nobody knows how to implement the machine learning techniques or neural network in predicting the B-cell epitope. That was a major challenge actually. So we make certain assumptions, and based on these assumptions, we develop the method for predicting B-cell epitope, and we achieve the accuracy something like nearly 66% actually. Though it is not very high accuracy, but seeing the other methods, physicochemical property based method, this is much better than the existing methods actually. And this method later become the standard in the field. Everybody is used for B-cell approach prediction. Same, we have the tools for confirmation of B-cell approach, but this method is designed from the sequences only. Most of the method they require the structure in order to predict the confirmation of B-cell approach. But we develop the method which need only the sequence. And from sequence we predict the confirmation approach. Same way, this is an updated version of the ABC PRED, latest version. This is another interesting paper or software we have. Most of the methods in the literature, they only predict whether it's a B-cell epitope or not, means the peptide which can induce the B-cell or not. Actually. But they don't tell what type of antibody will release actually. This method tells that whether this peptide will release IgG type of antibodies, IgE type of antibodies, or IgA type of antibodies. So that is a further classification. The type of antibodies will be released by a peptide actually. Okay. This is for the innate immune system, because the innate immune system is an important part we have to understand. So we created the database for the pattern recognition receptors basically. This is available. This is another part, innate immune system. We can identify the peptide-based vaccine adjuvants. Peptide-based vaccine adjuvants. So we have developed the method for peptide-based vaccine adjuvants, nucleotide-based vaccine adjuvants, and RNA-based vaccine adjuvants. So these are the different type of adjuvants we develop, and this is based on the TLR recognition. Because this is important. Most of the bioinformaticians thought that if they know the computer programming, then the problem will solve. No. You have to understand immunology or biology in detail in order to develop any method. Then you can develop the methods for that problem, basically. So here is a method where they help to design the vaccine adjuvants actually. That is called simply in the drugs salts actually. Okay, so here is a peptide or RNA or DNA based vaccine adjuvants. Because nowadays there is a problem of allergy actually. A lot of genetically modified foods are there actually. Okay, we don't know whether the particular protein which is introduced in the market is allergic or not actually. So this method predict whether a given protein is allergenic or not, as well as IgE epitope. IgE epitope is directly related to the allergy actually. So this is a summary of the tools we develop in our group for different purposes. Let's say T-cell epitope. These are some of the important tools. I'm not covering all the tools from our group, only covering few tools. If you are interested in a B-cell, these are some tools. If you are interested in the vaccine adjuvants, basically, then these are the tools actually. So these are the different type of tools which is available from our group. You can use them actually. Beside this, because in vaccine you are normally predicting the epitope or antigenic region which can serve as a immunotherapy or as a vaccine candidate. But there is another thing actually. What is the half-life of the peptide? That's another important part basically. So we have the developed the software which can predict the half-life of the peptides in given, let's say, in different body fluids, basically, in blood actually. As well as you have to predict toxicity of these peptide or protein, because they may be toxic to the body actually. So we have the software called Toxin Pride actually, which can help you to predict toxicity. Believe me, this year, due to the coronavirus and other things, the Toxin Pride, the server, is heavily used, cited more than 170 times actually in last few months, basically. Then there is another type of toxicity, that's a hemolytic peptide identity, means they lyse the RBC cells actually, because they are a different type of cells. These peptides, they can lyse the RBC. So we cannot use them as a vaccine. So there should be some server which can help you to predict. So this is for toxicity prediction. Then these are the examples, some portal we have given where you can work. Let's say this portal is for tuberculosis actually. If you want to design the tuberculosis vaccine against any strain, including new strain, because sequencing new strain is not a difficult task, then this portal will help you to identify the vaccine candidates. Same time, let's say a few years back, Ebola virus was there actually. So we developed this platform just to help the biologists. We predicted the tools, we predicted the potential vaccine candidate, drug candidate, diagnostic candidates, so that you can use them from our platform actually. Same way we did for the nowadays concept of the vaccine. I'm not going into the detail because these are the different type of trials actually. Okay, or some of them are already in the market. So we point out that there are chances that the vaccines may have some side effects actually. So be aware about them actually. And we also suggest the possible solutions to check their toxicity and the region of side effects actually. This opinion paper we published a few months back, and we already work on the one cytokine which is basically responsible is IL-6. Because IL-6 is responsible for interferon gamma. We have already developed the method earlier actually. So recently we developed the method for IL-6 also, so that it will predict whether it will induce the IL-6 or not actually. So this is a server. If you are interested, you can use it. Believe me, we have the more than 250 web servers which are available to the public. So if you are interested, you can use them actually. All are free to the public. Okay, the type of information is stored here. This is a web page. You can use it if you are interested in the coronavirus actually, and you can get all type of informations. What is a genome, genomics, related informations, what type of genomes have been sequenced. So what we have done actually, we have just collected and compiled the information from the literature. So we are not there generating the data. We are just collecting and compiling the information from the literature so that user can use it actually. Same way there is a this menu allow diagnosis. Let's say what type of diagnostic tools are there. We predicted the different type of primers actually for predicting the disease actually. And then we have developed the immunotherapy tools, the possible peptides which can serve as a vaccine candidate. Same, there is a drug design software, structures of these targets, everything is given here. So the overall aim here actually, we are collecting, compiling, and predicting the information related to the coronavirus, so that anybody who is working in this area may get the help from our portal. We don't need any credit or anything. Anybody can use them actually. They are all open actually. Thank you very much. I think I am within the time actually. Okay, thank you.
Thank you very much, sir. That was such a wonderful talk. And I now would like to request Dr. Anjani Devi, Vignan's Foundation for Science and Technology Research, to introduce the next speaker. Ma'am, please.
Good morning, everyone. I take immense pleasure in introducing Dr. Prashantyan Suravajhala. Dr. Prashantyan Suravajhala is Dr. N. Prashant Suravajhala is Senior Scientist at Birla Institute of Scientific Research, Jaipur. He obtained his PhD degree in systems biology. He went on to gain more than 7 years of postdoctoral experience across 4 different laboratories. He has interests exploring the known unknown regions in human genome, primarily working on the top-down systems biology approaches of hypothetical proteins. For the last 5 years, he has developed an interest in long, non-coding RNAs in humans, elucidating the mechanisms underpinning small molecular interactions through clinical exomes. He founded bioclues.org in 2005, India's largest bioinformatics society, which advocates open access. The organization aims to bridge mentor-mentee relationships and has been successful in mentoring as many as 2,000 graduate students across 30 countries. He is also associated with bioinformatics.org and a host of other humanitarian organizations like Rotary Community Corps. Now, I request Dr. Prashant to deliver his talk on Systems Genomics of Prostate Cancer, Indian Perspective. Sir?
Thank you so much, Dr. Anjani, for the kind introduction. Is my voice audible, please?
Yes.
And screen visible? Yeah. My internet connection is a bit unstable today. I don't know why, but I apologize for this. I hope it is audible. Yeah.
Yes, sir. It is audible, sir, and your slides are also visible.
Yeah. Thank you. Yeah. So at the outset, I would like to congratulate and thank InSyB organizers, especially Professor Kavikishore sir, Doctors Rao, Anil, Jalaja, Abhinav, and Tarun Pal for graciously inviting me for this particular conference. InSyB has been a wonderful conference series, especially this one. It's been about a conference with over 600 participants, which is quite amazing. I feel greatly honored, to deliver this particular talk after GPS Raghava, who is the doyen of bioinformatics in India and also the past president of BioClues. Thank you very much, sir, for this wonderful talk. So my particular talk probably, would delve on the history and rationale behind the prostate cancer project. So where are we now? I'll just, try to summarize the things. And I'll also talk about the kind of problems and challenges that we are probably looking into these directions, and the whole exome sequencing spectrum of prostate cancer, which we have started in relevance to India, and the kind of inherent pathways, and the big data challenge and the machine learning heuristics that we are just trying to attempt. We have just started attempting it. But into COVID times, we couldn't finish it. And then I'll also talk about the long non-coding RNAs and the regulatory elements associated with this. So I come from Birla Institute of Scientific Research, which is a sister concern of Birla Institute of Technology based in Ranchi in Jharkhand. Our current chairman is C.K. Birla, who is at the bottommost photograph here. And ours is a philanthropic institute, just like Tata's and Birla's. And we have got a state-of-the-art facility based in Jaipur with 2 departments. One is biotechnology and bioinformatics, and the other one is the remote sensing. We also have got a planetarium, state-of-the-art museum, and a big auditorium. And our current director is Dr. Punendra Ghosh, and Dr. Krishnamon is our head R&D, who is also my mentor. We're a group of 14 scientists, 7 PhD fellows, and 2 postdocs, of which 6 PhD fellows are working with me. So before I set up this particular thing, so I just try to tell why this prostate cancer project came into came into our mind, came into the objective hypothesis that we have set. So it was way back in 2013 and 14, during my third postdoc heydays. I've had an illustrious career of about 7.5, 8 years of postdoc, and I've worked across 4 different laboratories. So during my third postdoc in 2013, I just returned again back to Boston and Boston to Denmark with a brief stint in India during that time. And my postdoc mentor, Haja Kadamidin's lab, they were working on something called feeding deficiency traits. So they would probably overfeed the mice and overfeed the eutherians, like cattle, buffalo, and all those animals were sitting in the animal genetics labs, and they would try to look what are those particular trait specific genes that are probably associated with those particular animals. So, like as you could see here, there's a mice. This was the earlier photograph. It is overfed and it becomes obese. And likewise, the cattle as well. If you just keep if you keep raising cattle, it will keep on eating and eating and eating, and the farmers would probably think that it would probably eat more milk. But one thing, happened, when these animals are really overfed, the 2 PhD fellows in that lab, when I was doing the postdoc, their job is to feed it, collect the serum, from the, from those animals and try to see the differential expression count in those particular animals and see if there are any genes, that are upregulated or downregulated. So in nativity, if you really see, if you really eat normal, we assume that we are healthy. If we eat less, we are unhealthy. And if we eat optimal across different time periods, giving some breaks, again, we are healthy. But if we are eating too much, the unfortunate thing is we become unhealthy. There are a lot of unnecessary metabolites prevailing in your body that might make you obese. This we know, but we didn't know whether or not those animals would become prostate. Those would be affected with prostate cancer, and that was how this particular project started. And I thought if there are any obesity related genes that are associated with prostate cancer. And then after I had another postdoc between 2014 and 16, and then I just sort of coming back to India, and that was how I started writing a grant. And this grant is a major grant of 1 crore, which I gotten through Novartis and BrainPan, that I'm associated with it. So this particular prostate cancer, we just asked if there are any mutations relevant to India. That's because 1 in 9 men are diagnosed with prostate cancer during their lifetime. It's usually the incidence is above remarkable, and with an age factor precedent set above 55 years. And it is the second leading cause of death globally, and it has become third most prevalent cancer after breast and oral cancer in India. And it's rapidly growing. There are different things. I mean, the life expectancy is going to be declining, and there has been a declining fertility rates as well. Although at this point of time we are only looking into the so-called somatic mutations, not the germline mutations, because we haven't really understood what are the mutations specific to prostate cancer in Indian subpopulation. So this is how it is. The take-home message here is, as for the Globocon statistics for 2018.
Sir, your voice is not audible. Could you please unmute yourself, sir?
Yeah, can you hear me now? Yeah.
Yes, sir, you're audible now.
Yeah, someone
Please go
muted me. I'm really sorry. So can you see the screen as well? Yeah.
Yes, sir. Yes, sir. The screen is visible.
So and again, this prostate cancer is, quite seen in African Americans. It's because, it comes with the diet factors as well, because probably they eat a lot of red meat. Whereas Asian Indians, many Asian Indians are comparatively vegetarians, if not vegetarians, or probably at least, people eat, fish when compared to the red meat. So we Asian Indians eat a lot of, broccoli kind of things. So we are not, that inherent to, prostate cancer. But if you really see the far west, where, there's a lot of, meat eating people, especially in the Americas, so prostate cancer is absolutely visible over there. So, so if you really look into the risk factors, so there's always an association between fat consumptions and they could be also be associated with countries with greater per capita milk consumption. Okay, so the most important thing here is there is no association between smoking and alcohol. I mean, it is assumed that if people smoke a lot, maybe people drink a lot, they might be associated with cancers like prostate. But just imagine a person who is getting a prostate risk at the age of 55, and maybe you would have really started drinking right from his age 21. He would not have really gotten just cancer just because he's over drinking or he's over smoking. So that assumption is absolutely a kind of a false entity. And of course, low per capita means countries with low per capita has got lowest risk of prostate cancer, just like in India, or probably some of the African countries. But there's also an assumption that in Americas or Australia or UK, they have got higher risk of prostate cancer because they've got high per capita income. And if you really see the detection things, detection is usually done using prostate serum antigen and distal rectal examination. There's also something like biopsies. There are also some markers that are usually done in the pathological lab, like the kallikrein HK3 markers, which is a human kallikrein gene. And there are also tumor markers. There are also some tumor necrosis metastasis factors, which is what we call it as a TNM grading for any cancers. But here for prostate cancer, there's something called Gleason score classification. So if a Gleason score is above 5, then that indicates that the prostate cancer risk is higher. And below 5 is nothing but they're all the benign tumors. And there are some urinary biomarkers. One is a well-known long non-coding RNA called MELET1, which is well studied. So what we have done, when we came here was we have attempted to write this particular grant to DBT, and unfortunately it was not true. But I thought, since I was associated with BrainPan, we tried to contact Novartis and other companies. And that is how we got this major grant. So we started with a pilot of 10 samples that was escalated to another 10 samples, about 20 samples, and another 110 samples are on the angle, the sequence. So what we did was a whole exome sequencing of these 10 samples with about 5 benign cases and 2 with intermediate Gleason scores and 3 Gleason scores about 7. And the ethical clearance was judiciously taken from the Rukmini Birla hospitals, thanks to our clinical collaborators. And these are the cases. We have taken an inclusion criteria of age above 55, of course, with a Gleason score of 6, with a prostate serum antigen of 4 nanogram per ml. And these are all the malignant. There's something called benign prostatic hyperplasia, where the control samples have got Gleason score less than 6, as I told you. And if you really see, we have developed a lot of, good number of, bash-based pipelines to benchmark these whole exome sequencing pipelines. So you simply give a FASTQ file, and then at the end of the day, you'll get a lot of variants with SNPs that are associated with different disease conditions. So we have benchmarked this pipeline, which is MENA et al. 2018, which is highly downloaded protocol, which was widely used by more than 25,000 users globally. And this is how the classification is. We got a number of reads. with a good size across, and all of them were quality checked. And if you really look back into the basic nitty-gritties of these particular samples, out of these samples, all samples are all patients are all vegetarians actually, and yet we were a bit surprised, how come many of these particular samples, although being vegetarian, they have got the prostate cancer risk. So if you really talk about the results, we asked that there are some high impact variants across high grade prostate cancer tumors that are specifically inherent to Indian subpopulation. So if you really see, we found about 30 causal genes that are responsible for prostate cancer in India. So some of the well-known, BRCA1 and BRCA2, which has got, which are also associated with breast cancer, that are also seen in men, as you could see here. There is also a CTNS gene, which is associated with vitamin D factor. Although we receive a lot of sunshine, even many of these prostate cancer patients have got this CTNS mutations sitting over here, and also some PUSH2A and XP mutations. These are the DNA mismatch repair proteins. There are also some proteins like GMP-TAB, which are involved with transcription factors as well. And of course, the TP53, which is a tumor suppressor, which is also associated with obesity. The COL6A1, which is primarily a copy number variant, where that particular chunks are also seen across different genes. That is also associated with obesity as well. And we have also found some of the other genes that are associated with mitochondrial function as well. So OPA1, which is a well-known mitochondrial gene as well. So which means it's also probably inherent, because there could be mitochondrial mutations that might be transcended from the mother as well. So OPA1 is one of those particular victims. So as you could see, the square boxes are all those genes that are seen in Caucasian or West, and the ones that are in circle, these are all those mutations that are primarily seen in Indian conditions. So this shows that in India, diabetes is also associated with prostate cancer risk. And this is what we have inherently done. We have checked across a union of intersection of variants between cases and controls. And we have done some proved statistics. So these are our first authors, Ayam Gupta and Nidhi Shukla, my PhD fellows, who have done this particular work amazingly, and which we recently published this. So the part one of the conclusions is our patients in our cohort are vegetarians, and they are yet prostate victims. Our pilot study revealed that there are mutations associated with obesity, diet, mitochondrial dysfunction, and as well as vitamin D deficiency. There are some tumor suppressors. There's also a copy number variant like COL6A1 detected. You might ask, in a whole exome sequencing pipeline, usually you cannot identify copy number variants, because copy number variants are bigger chunks of mutations, anywhere between 40 bases to up to 1500 bases long. And they're all those segments, that are seen across, different genes. So usually a whole exome sequencing cannot detect it. But we have tweaked our pipeline to identify some of these copy number variants. Some of the copy number variants are seen. And there are some diet-induced obesity genes, like these three, and also CTNS gene. And we have come to a conclusion that yes, we have developed a conceptualized framework to prioritize certain mutations for prostate cancer risk in Indian subpopulation. So that project was the part one. And I've been, somehow interested in long non-coding RNAs because, my PhD, I formulated something on the hypothetical proteins. Thanks to my wonderful mentors, we have developed, some databases and some platforms to identify whether or not a hypothetical protein, can be, classified in a good way or not. But eventually, many of these hypothetical proteins have turned themselves to pseudogenes, or some of them were misannotated as long non-coding RNAs. Okay, and that was how I tried to ask if there are any known unknown regions in the genome, that can be essentially tagged as long non-coding RNAs, and maybe who knows, long non-coding RNAs can also be associated with prostate cancer. So this is what we try to walk through the genome story. So if you have any gene somewhere here which is associated with say your diseased phenotype, so in this case a prostate cancer. So this is an ensemble genome browser where this entire region is a gene responsible for prostate cancer with a contig like this. As you could see here, there are some yellow blocks, which are in fact enhancers, as you could see here. Okay, and there are some blue ones which are the CTCF motifs. Okay, and there are some squared boxes somewhere over here which are the long non-coding RNAs, as you could see here, these ones. Okay, so which means if you have a hypothetical, if you have a gene of interest for any diseased phenotype, if you walk through the genome, usually the 5' untranslated region, up to 5,000 region or 5,000 bases of upstream of that particular gene, you will find these regulatory elements which might influence the way that particular gene works. So they might ensure that just because there is a regulatory element sitting over here, this regulatory element might have an influence on how that particular gene might harbor certain mutations sitting in it. Okay, so this is what we call it as a genome walking. So this is what a BRCA2 gene, and incidentally, Vignan University students are also doing this particular genome walking story along with Nidhi Shukla. So this is what we are trying to work. And a visiting graduate student, Narmada, she has performed this particular genome walking story as well. And we have identified some long non-coding RNAs and long immunogenic non-coding RNAs as well. This is still ongoing. We have just characterized some of these long non-coding RNAs, which we are still trying to attempt and come to a consensus and ask if there is any long non-coding RNA, that could also be associated with these diseases. So this is a long non-coding RNA, non-exact 053810. But eventually, you might even ask, Prash, why is it you're always talking about long non-coding RNAs? So there could always be some other small non-coding RNAs like the pvRNAs or the microRNAs as well. So microRNAs, although they're pretty short, they would fall short of mutations because they're highly conserved. They're anywhere between 18 to 24 bases. So being 18 to 24 bases, the chance of having mutations in them is very less. So we try to ask if there are long non-coding RNAs, and eventually there are also some pvRNAs. pvRNAs are anywhere between 24 bases to 45 bases in length. So we have also found this pvRNA 3359922 as well as of now. So we are just still working on that. Nidhi and I they are working on this. So and something like exciting popped up in between. So this COVID came and all our lab activities have really stopped. And then I started working on this SARS-CoV-2 along with Keshav Singh. So what happened was when this COVID came into picture, we again asked if there are any receptors sitting somewhere in the host genome where in addition to the ACE2 and TMP-RSS2, which are the receptors in the host that invite SARS-CoV-2 to enter into the host, are there any long non-coding RNAs that are probably sitting over here? Are there any transpressed, or are they really sitting over in the host that might be associated with this SARS-CoV-2 as well? And then we instantly, from the data that we have observed, that we have found that this non-exact 23988 is highly expressed in prostate as well. And this long non-coding RNA is in pursuit of this particular receptor-mediated entries. There are also other long non-coding RNAs that are substantially expressed. The reason why I'm talking about this long non-coding RNAs is because they just sit in the host, they invite SARS-CoV-2, and the long non-coding RNAs with the help of ACE2 and TMP-RSS2 try to receive it, and as soon as they receive it, the hijacking process of SARS-CoV-2 comes into shape. So it tries to eat away all the mitochondria, and that's one of the reasons why people will not be able to breathe properly, and that results in cellular death and finally asphyxiation, and finally the person dies. But in milieu, there is there is most importantly in a lot of prostate cancer associated long non-coding RNAs in this particular process. So the part 2 of this particular story is there are some pVRNAs also in addition to the long non-coding RNAs. We try to identify whether or not these long non-coding RNAs have an influence on this particular gene, whether or not they interact with the proteins. And we are just still trying to identify if any of these long non-coding RNAs or pVRNAs could really make up a good story here, where they could be associated as interaction partners. Thereby, we could develop a kind of a viable, prognostic marker towards development of prostate cancer risk in Indian subpopulation. And recently, what we have developed is, apart from this hijacking story, we have also come up with a new hypothesis called transgression hypothesis. This manuscript is communicated. We are hopeful to get an answer sometime by this year end. So there is a non-structural protein 15, which is over here, NSP15. So what happens is this non-structural protein 15 has got a highest chance of transgressing the long non-coding RNAs, because if you simply blast this NSP15, it shows resemblance with a lot of non-HSAT 255167. And what happens is, as you could see, these are those particular bases that are absolutely transgressed. When I say transgressed, this long non-coding RNA is sitting in the host. NSP15 takes control of the virus and the host. And in the milieu, the long non-coding RNA bases are eaten away by these viral particles. And that is how this CTNND1, which is a gene called connectin, ketenin. This particular ketenin gene is also known to be associated with this long non-coding RNA. And this is how the transgression hypothesis unfolds. So, what happens is there are also other receptors in addition to the transmembrane protein RSS1 and ACE2, angiotensin converting enzyme. Is other receptor like interleukin 17, which is sitting over there, where these long non-coding RNAs are being controlled by these particular receptors. Okay. So what now? This is my final part of the story. So now we have annotated a lot of genes. We have identified some mutations across 30 genes, out of which about 7 genes are primarily responsible. A couple of them are associated with diabetes. A few of them are associated with mitochondria and all. So can we, come up with something like a predictive analytics? Can we come up and say that yes, we have identified some genes responsible for prostate cancer risk. Now, what if we have some population, that is coming up in the near future? Can we say that yes, these are the mutations that might be associated in this given subpopulation if we have a patient coming with such disease phenotypes. He might be obese, he might be prostate cancer risk, he might be associated with a mitochondrial dysfunction, and if he has a scrotal pain, can we just by looking at his vitals, can we really say that yes, he might harbor these particular mutations? So we're trying to look into the precision scale. That is what we are trying to ask. And so we have a whole exome sequencing data. We have a list of variants over here. And in part 2, we have got all the regulatory elements, the long non-coding RNAs, and also the RNA-seq, the whole transcriptome sequencing data, which we are currently doing along with. And now the part 3 is a deep variant approach, which we have attempted it. Our other program is still running, which we are just trying to ask if we can combine this and come up with some predictive analytics.
Sir, there is some glitch in your voice. Can you please fix the problem?
I'm sorry for this. I think I've had a very poor connection. So yeah, I hope you can hear me. Yeah.
Yes, sir. Yes, sir. Now you are audible.
Yeah. So you can also see the screen, right? Yeah.
Yes, sir.
Yeah. So so this is what we try to ask, and then there are no diagnostic methods that are available in the wet lab today. Even if they're available, the diagnostics would be very much costly. It's not a kind of an easy thing treating a kind of a prostate cancer patient. It involves a lot of monies as well. So as you could see here, the costs are absolutely very heavy here. Even in India as well, right from biopsy till getting the digital rectal examination done, it takes anywhere between 20,000 to 35,000 rupees, which is pretty expensive. So since this is a largely funded project, coming from BrainPan and Novartis, so we thought of coming up with a kind of a kit. Can we develop a kind of a simple pregnancy like kit where because many men are usually stigmatic, they don't usually come and open up and say that they've got some pain in scrotum or they've got some pain in rectum. So can we come up with a high turnaround time kind of kit where with the simplest urine they could probably check whether or not they are at prostate cancer risk. So although MALAT1 is well-known urinary biomarker, it is not associated with prostate cancer. It is associated with genitourinary cancers. So we are just not trying to attempt. We are just not trying to ask if we can really come up with a new cancer-based prognostic kit. It can even be a kit. It can even be a kind of an NGS panel, which is pretty cheap. So simply taking up and checking from those particular measures whether or not those particular genes, if they're present in blood samples, then we can say that the person is prostate cancer risk. So so the final lessons and final summary over here is We have identified about 10 long non-coding RNAs and 1 pbRNA. Although we have found about 10 long non-coding RNAs, we have jotted down to only 3 at this point of time. Our analysis is further leading to understanding the regulatory elements, the CTCF motifs and the promoter flanking regions, open chromatin elements, the enhancers and the transcription factor binding sites. Our present results, probably support the multi-gene panel testing because we might have many genes where we might take the coding genes, the non-coding genes that are associated with disease phenotypes like prostate, genitourinary cancers or whatever, and come up with a kind of a panel. But we need some kind of a deep variant approach. We need some machine learning approach which we are trying to work on. So that would probably set a precedent to work on the immediate goal, like taking the deep variant based approach, taking the Gleason score attributes, and the distant goal would be to look into the hereditary conditions. Say, for example, in India, if a person aged 55 is prostate cancer risk, what is the percent chance that his 26-year-old son might also be associated with prostate cancer? Okay, when he turns 52. So that kind of things we are trying to ask, and we are trying to simply work towards these particular conditions. And we also would like to look into the role of factors like diabetes, obesity, vitamin D, and other dietary elements. Where there are some genetic factors that are associated as of now. There are some Indian specific variants as well. There are some genetic changes that are responsible for this prostate cancer as well, like the CNVs and the PON mutations. Of course, we're also looking into the long non-coding RNAs like MALAT1, taking into this thing as a picture. And we are also trying to ask some questions. What if a benign prostatic hyperplasia patient who has never been considered as a prostate cancer victim, what is the percent chance that he might be prostate cancer risk in the future? And what happens if a person who is with hemorrhoids, who is down with fistula, who is down with piles, what is the percent chance that he might also be prostate cancer risk by the time he turns 55? Okay, so these are all the predictive analytics that we are still working on. Unfortunately, in India, we are still in infancy at this point of time. So I think I'll probably end here. I'll be happy to take some questions here. But before I end, I would like to thank Kavikishore sir, Dr. Srangarao, Anil, Jalaja, Binav, and Tarun for inviting me, and also Asif as well, who has been graciously helping BioCruise as well. This work would not have been done without these two wonderful people. Nidhi, who is a women scientist fellow and also a PhD fellow. I am Gupta as well. Mamata and Narmada, they were graduate students who worked, and now Womandeep, who is my another PhD fellow, where Sugunakar is the main supervisor. She's starting working on prostate cancer. And these are the other PhD fellows who are working on different projects like vitamin K and the known unknown regions. Sanchita is also here, and Sharini working on the vitamin K. These are all the clinicians. In fact, clinicians' role is very, very crucial in India because without them, we would not have gotten the samples. My full credits to Devendra Sharma and Manish Vijay, the clinicians, urologists and pathologists working over there. Amrit and Saurabh, through which we got the brain pan funding through Novartis of 1 crore. And of course, Ashwin, who is my co-PI. And Krishnan sir is my mentor. Obal, my collaborator, and Dr. Praveen Mathur, who is a well-known pediatric urologist. And of course, Kavikishore sir and Giri sir, who's whom we cannot really forget. They have been absolutely wonderful people to back with. And Vikram Singh Chauhan is the one who ensured our ethics clearance were in place, and Jyotsna Patra as a part of a practical consortium, and Nirmal Lohia sir, who has been a wonderful person. And Subbu, my collaborator, of course, and Vinod Nigam, my collaborator. So thank you so much. The most important thing, the bottom line, I'll just take just 30 seconds here. There are about several people attending here. I know we have really faced a Herculean task of 2020 this year. I request all of you to learn to collaborate, not to compete. This is the time where we all come together, join hands, join forces, come together and say that, we are Team India. We are all, working towards, for development of science. If we start thinking that, the other person is, competing with each other, probably we should also compete, then that's absolutely a wrong thing. And that is where, these 3 C's, come into picture. Always, try to collaborate, try to converge, try to learn from each other's, expertise. And that would really bring out, a wonderful, consistency. And that is where, we can probably, combat, COVID-like, viruses in the community. Thank you so much. Thank you.
Thank you, Dr. Prashant. Now, the session is open for discussion. Participants.
I have a question. This is Kavi Kishore.
Yeah, please, sir. Yeah, thank you, sir.
In 110 samples that you have taken for prostate cancer analysis,
yes, sir.
You said, most of them are vegetarians, but still they are getting, the prostate cancer or at the risk of prostate cancer, How many samples you have taken from the non-vegetarian group?
Sir, that is where, in fact, our study is a very pilot study. So this sample is only from 20 samples. The study is only from 20 samples so far. The other exome sequencing is still being done for about other 20 samples. So total about 40 samples we are going to do now. Out of 20 samples for exome sequencing that was done, 19 samples were vegetarians from our study, from our understanding, when we counsel them. And one other person is a non-vegetarian, that is from our understanding. So this is a kind of a very beginning to be understood, sir. It's only, a very crude, thoughts, going through our minds. What would happen, if there are non-vegetarians that are really obese, and what would be their fate, if they're really a prostate cancer risk? So, yeah.
The long non-coding RNAs and pVRNAs that you have identified with prostate cancer, these non-coding RNAs and pVRNA have also been identified in other countries, in other groups?
No,
or
sir.
Only in India?
No, yeah, it's only with respect to Indian subpopulation. So, in fact, we have seen these long non-coding RNAs only relative to Indian subpopulation, but one long non-coding RNA, which is 2399AAA, that is from BRCA2 actually, that is present somewhere in the upstream of this BRCA gene. That is quite common, and that is rigorously seen across other patients as well. But sir, this is a kind of a walking story, which is a genome walking story, which we have identified from a whole exome sequencing. So we are not sure yet whether or not this long non-coding RNA is differentially expressed from the whole transcriptome shotgun sequencing where our study is underway. So we have given these particular samples to our service provider. So once we get those RNA-seq data, it will be wonderful to see if those long non-coding RNAs are differentially expressed in those genes. So that is where, we are trying to begin, trying to understand.
Yeah, and also I have a foolish question to ask.
Yeah, please, sir.
Whether it would be possible for us, to use some of these long non-coding RNA markers or, the pVRNA for predicting, the prostate cancer. Can we use it as a diagnostic tool to predict, in advance, ahead of times, to the prostate cancer?
Right. I mean, this is a wonderful point. So it's not at all a foolish question. That's where, we are trying to understand. So there's something called human kallikrein markers, which there's a gene called HK3 that is well known for prostate cancer or, rectal risk. So what we are trying to bridge the gap here is, say, let's assume that we have identified a long non-coding RNA that is absolutely responsible for prostate cancer. Now we are going to ask whether or not this long non-coding RNA is circulating in the blood as well, or are they really associated to be considered as circulatory biomarkers? That is number one. And if they are circulating, then there is a chance that these circulating biomarkers can also be associated with the pathologic manifestation like the human kallikrein gene. So then we would try to ask if any of these long non-coding RNAs have an influence on some kallikrein genes like HK3, where they can be subjected to development of some prognostic markers. So this is a very the study is absolutely in a kind of an infancy, sir. We might get some exciting results sometime by the end of next year. because we also have a very good plan to come up with a development of a good kit to Novartis and BrainPan, where the study is underway. Because it's largely funded, and the current problem that we are trying to face is the lack of samples. Unfortunately, we are not really getting samples as of now. But your question is absolutely very valid, sir. Thank you for asking.
Yeah, thank you very much, Dr. Prashant. I have thoroughly enjoyed.
Hello, Prashant. One question from Professor Krupanidhi.
Oh yeah, please, sir. Please, sir. Please.
Yeah. So Prashant, I've been following your talk. It is quite interesting, and I wanted to get to know, because you mentioned 10 long non-coding RNAs. So they're, as I presume, I may be wrong. So they are only prognostic markers, not causative features, no?
Yes, sir. I mean, as I told you, these are all those long non-coding RNAs which we presume that they have identified or they are associated with the genes responsible for the whole exome sequencing study.
Yeah.
So, in order to make it further, we need to validate their expression. We need to validate their regulatory role and the differential expression from the RNA-seq data as well. So the RNA-seq data for about 6 samples is underway right now, and we might do RNA-seq for another 20 samples sometime during the next few months, sir. So once we identify these long non-coding RNAs from these differential expressed count as well, then we can say that yes, out of these 10 long non-coding RNAs, maybe 2 or 3 or handful of them could be associated with prognosis. Yeah.
So that can be linked from the childhood onwards.
No, these are all the somatics, sir. I would say. I mean, unfortunately, we cannot say, because, and of course, I mean, if we really say that might really come up with childhood, that would, probably be a question of yes or no. That's because we don't know whether or not any of these particular mutations are seen in these long non-coding RNAs as well. That we need to identify only from RNA-seq. Although genetic variation is seen in the long non-coding RNAs, the risk of attributing the mutation specific to long non-coding RNAs is very minimal from the whole exome sequencing study. Yeah. Because from the whole exome sequencing, we'll not be able to identify the long non-coding RNAs at all. So it's only
these long non-coding RNAs will be having influence on the causative genes.
Yeah, very much. That's where these long non-coding RNA protein coding gene interaction studies are underway, sir. So so that's what we have tried to attempt. Yeah.
Is there any transcriptomics coming out of non-coding RNAs? That is non-coding gene, that SYN and line.
Yes, sir. That is where our studies are underway. Our samples are being analyzed now for RNA-seq, the transcriptome study. So we are looking forward to see their expression in these particular samples as well.
Yeah, okay, thank you. There's a lot to know through you. Thank you.
Yeah, thank you, sir. Yeah, thank you.
Sir, any more queries from the participants? Okay, if not, thank you, Dr. Prashant. You have given us a clear-cut idea on the prostate cancer, reasons for prostate cancers. Besides, you have given us the idea about what are the risk factors associated with the prostate cancer. Thank you, sir. Thank you for your enlightening speech.
All right, sir. That shows a mere brilliance. And now I'd like to request Ms. Pooja to present over here the next speaker. Ms. Pooja, please.
Dr. Mohammad Asif Khan is an associate professor at the Perdana University School of Data Sciences, Malaysia. He is also a visiting professor at Bismialam Waqif University, Turkey. In conjunction with his induction to the International Fellowship for Outstanding Researchers, program by the Scientific and Technological Research Council of Turkey, Dr. Khan obtained his PhD from the National University of Singapore. His research interests are in the area of biological data warehousing and applications of informatics to the study of immune responses, vaccines, viruses, venom, toxins, drug design, and disease biomarkers. He has authored more than 50 international peer-reviewed research and review articles, published in various international journals, books, and conferences, and presented his work at numerous international conferences. He has been involved in the development of several novel bioinformatics methodologies, tools, and specialized databases, and has 3 patents granted. He is currently the elected president of the Asia Pacific Bioinformatics Network. He has done various important roles in the organization of many local and international bioinformatics conferences, meetings, and workshops. Now, I request Dr. Mohammad Asif Khan to address. Sir, please.
All right. Thank you. Thank you for the kind introduction. Can you hear me? Can you give me a sign?
Yes, sir, we can hear you.
Okay, and the screen is visible?
Yes, sir.
Okay, wonderful. Thank you so much. And first of all, I want to thank the organizers for inviting me to give a talk at InSyB 2020. It's a pleasure to be here, and I'm very happy to see how InSyB 2020 is shaping up. Also representing EpiBioNet, and this is one of our annual events. I thank Prof. Kishore for agreeing to helm this responsibility despite not being given enough time. He managed to pull this off, and really happy to see the number of registrants, 900 something. So, and many of them are joining today. And I also want to thank Prash for recommending Prof. Kishore to propose hosting InSyB. Overall, thank you, a big thank you to VFSTR University for all the organizing that you have done wonderfully in order to bring the community together for this year's InSyB. Okay, in the interest of time, I will proceed. Okay, so the title of my talk, as you can see, so I'm not going to repeat, but essentially, this is actually the work of my master's student, Chong Lee Chuen, sure of her later. And you can think of this project as trying to, compress data. It's more of a data compression project. You have used WinZip and others. The only difference is this is for biological sequences. So that's a context that I can provide you at the moment, but let's move forward. Now, this is a picture I think many of you may have seen given the current corona pandemic. And this was a vivid reminder that, corona is not a new pandemic. Well, it's a new pandemic, but pandemics have been part of our history. And we have had a number of them that have been recorded since 165 AD. So we have the Antonian plague that affected 5 million people with a death toll of 5 million people, and many more along the way. A number of these are viruses. We can see that the yellow fever is there with 100 to 150K. HIV/AIDS, which is still a problem. 25 to 35 million people get affected. Spanish flu was a big one in 1918, and that killed, based on estimates, 40 to 50 million, and some estimates go even higher. And we had few smaller ones, but every life matters. So it doesn't matter whether they're small or big, but viruses have been part of our history. So with the spread of humanity, well, the threats are also there. Corona is right up there in the front line at the moment. This is an old number, and at the moment, I just quickly did a check on the estimates. We are at the tune of 1.7 million people who have died because of corona. And it's going to be a year soon since when we kind of, I think, identified it to be corona. Corona around January. So yeah, this is a sad state, and hopefully through advances in medicine and hopefully championed by bioinformatics in this era, we could do so much more to prepare ourselves for these kind of pandemics. All right, so now viruses are able to do this and become a threat to humanity is because of their ability to change very quickly, especially RNA viruses, as you can see here. Of course, compared to bacteria and eukaryotes, viruses are one of those that change more rapidly, and RNA viruses are right up there. The mutation rate is very high, and And of course, they carry much smaller genome size. So, and these mutations, I wouldn't use the word mutation substitutions, because you can have insertion, deletion, mutation, recombination, many things can happen. So the right word would be probably substitution. But of course, in this plot, we are looking at mutation. But in general, so these substitutions, are one of the main reasons that pose a threat, because of escape from immune recognition, among other things, which I will cover in a bit. But another thing to remind ourselves is that these RNA viruses, when they infect one person, you could start with a founder virus, starting virus. And within one individual, over time, very quickly, you can have a variant of these viruses within just one individual. Forget about the human population. Within one person, you can start seeing mutations or changes that are different from the parent virus. So, and this population of viruses within that individual are called viral quasi-species. So like quasi, semi-pseudo species. So that's how bad it can be. That within one individual, you can start seeing different viruses over a period of time. All right, so this is one of the main reasons, and one of the good news is that because of the small genome size, we actually have the opportunity to sequence them. Of course, we can sequence things very quickly, and because of their small genome size, there's actually an abundance of genomic data, sequence data available in public repositories. So this is publicly available. Anybody can access these and study them, and this is also what makes viruses favorable and of interest to a lot of people in studying them, given the data abundance. This is just a quick reminder that how fast the data grows. I'm sure we all know that data is growing rapidly, but this is some sort of a data that I asked my student to track, and we track this for a period of 1 year. And for protein, we can see that it has grown from 4.9 million to 5.8 million for a rough period, slightly over than a year. And so that's about 18% growth overall, but nonetheless, close to 1 million new sequences were deposited over that period. And actually, if you look here, this period ends at November 19, and today we are in 2020 and slightly more than November. And I just checked the data again, the number of sequences, we are currently standing at 7.3 million. So this kind of sums up that the data growth is really rapid, and we need to be able to quickly analyze these sequences as fast as possible. And similarly, there's also, of course, a rapid increase in the nucleotide sequence data. I'm not going to go into the details. All right, so given this availability of sequences and the substitutions and changes that viruses go through, I think we need to, I think we can say in a nutshell that there's a lot of diversity, viral diversity. And I wanted to just quickly break down that diversity into 3 contexts, so that which some of us may not recognize or may not differentiate those when we use the word viral diversity loosely. So there's the genetic diversity. This is simply the diversity at the genetic level, the DNA sequences. Now, so what is protein diversity? Well, not all differences at the genetic level change or translate into differences at the protein level. So that would be your protein diversity. So it's not equal, because you have synonymous changes at the nucleotide level that do not change at the amino acid level. So, therefore, protein diversity would be slightly smaller. In comparison of genetic diversity. Then what is antigenic diversity? Antigenic diversity is more related to the immune system, immune recognition. So all those amino acid changes that you have at the protein diversity level may not necessarily impact or affect immune recognition, especially if you're focusing in the context of certain HLA profiles of certain individuals. So maybe specifically for my HLA profile, not every change would be relevant to my HLA profile. So there is that, maybe smaller set of antigenic diversity. So I hope that gives you a little context in terms of the differences between genetic, protein, and antigenic diversity. Now, antigenic diversity is probably, among all the diversity with respect to immune system, this is maybe the most relevant one. I think it's the most relevant one that should be studied when we want to design vaccine or look at therapeutics prophylactic approaches against viruses. Why is that so? Because these diversity, these changes, result in escape from immune recognition. And of course, this directly affects vaccine design. So you could have had a vaccine that has been designed, developed, and if infection happens and the variant is different from the strain that was used for the vaccine design, then there's possibility of escape from that recognition. And this is a problem that we see with influenza. Influenza vaccines go through this annual reformulation so that the vaccine strains, the strains that were used for the design of the vaccine, match with the circulating strains that are going around. And these circulating strains change, so we got to ensure that they match. So there's also this concern about this coronavirus vaccine that have been released. How confident are we that these vaccines would be able to capture the diversity of sequences, of strains that are circulating around? And there was a news about a new UK strain that seems to be spreading quickly. So these are always some of the concerns, and so therefore it's important to study antigenic diversity. There's another thing that we also want to take note of, which is original antigenic sin. This is actually popularly reported for dengue virus. In fact, it was originally observed in influenza virus. So you could read more about it. I don't want to go into details, but the general idea is that you get infected by one and you develop immunity against it, and then when the next infection happens, the next variant is very similar, yet different from the first one. So your memory response gets called instead of a new naive immune response. You call the memory response because of that recognition, but it's not the same. It's slightly different. So what happens is you end up with something called immunopathology. Immune system is trying to get rid of the virus, and it's doing all kind of attacks and all that, if you loosely call attacks, and so you end up with inflammation and other things. So in some sense, you end up damaging the immune system and ends up damaging the host without really effectively clearing the pathogen. So there's this term immunopathology. So this can be a side effect of this immune escape. If you have a memory response and you call that memory response, which is not effective, and in fact it should be calling a naive response, because this is different strain from the previous one. So there's a whole different work there that you could read up, but that's another reason why we want to study antigenic diversity and ensure that we try to extract as much as possible from the data, especially when there's a lot of data available. What can we say about antigenic diversity of that particular virus? And hopefully that knowledge gets used when we design vaccine. All right, so in general, study of viral diversity, I think can be easily simply divided into two ways: alignment dependent, and of course the other one would be alignment independent. Alignment dependent is the popular one because it allows us to nicely compare the sequences of these viruses, and we can then, once we align them, we can look at the diversity that's there. So you get conserved, non-conserved regions. The problem with alignment-dependent approach is that it breaks down for very diverse viruses. So I remember we were studying HIV, and there were there's certain positions in HIV that are so diverse. It was really difficult to align those regions, and there were a lot of challenges that we had to go through in order to try to get the best possible alignment that we could. And the other problem with this is it kind of restricts your ability to study diversity beyond a particular species, because the further you go from the species, let's say from species to genus to family, even if the virus is conserved, because you're now studying different sequences under a genus, species under a genus or under family, the number of species just grow, and therefore to study them together collectively, it's going to become more challenging. Imagine trying to align all the family members. Can be really hard. Maybe for some viruses, for some families it's not too bad, but for a lot of families it can be really bad. And what if you wanted to go beyond family? Or what if you just wanted to analyze all known viruses that have been reported to date? That's not going to be possible with an alignment-dependent approach, because you would lose a lot of that conservation. So there's a limit to alignment-dependent approach. Of course, it's useful. We need this kind of approach, but we also need to develop alignment-independent approaches. And there is a lot of, I think, nice work that's going on out there. And so our work here is actually contributing to the pool of tools available to do alignment independent approach analysis. This has been, I think I've seen a lot of nice work for phylogeny with regards to this. But anyway, so I just wanted to highlight that there's this two different strategy when we want to study viral diversity. All right, now, so you could have all the sequences of the viruses that you want to study. You can align them if you're going by the traditional approach. You can't, I mean, you should not study every one of those sequences because not everything might be, especially if you're aligning protein sequences, you don't want to study every one of them because they all may not be relevant to antigenic diversity, as we talked about earlier. Because what might be relevant to the immune system may not be all of those amino acid mutation. Maybe some of them would be relevant. So what would be the best way then to map the antigenic diversity in your sequence that you are interested in studying? Of course, wet lab would be the best way to do this. To study and determine which one of those parts of the sequence are epitopes. And we know from papers that not many of those regions would be epitope. Typically from 1 to 5% is thought to be epitope. So you're going to spend a lot of money, a lot of time in trying to study all these sequences. It's just not effective because the number of tests that you would have to carry out is just enormous. And this is just for maybe one species. Imagine if you wanted to study all the viruses out there, it's just not possible. Right, we would need something more high throughput in order to deal with the big data. Even then, it's I think a computational approach is necessary before we go to wet lab. So there's wet lab would be the best, but there's a big limitation there. Of course, the next one would be in terms of computational approach. We could predict the epitopes. Could we predict the epitopes? But unfortunately, we don't have prediction models for the different repertoire of people's immune system. For example, my HLA profile might be different from others, and the last I checked, there are like 25,000 different HLA molecules that have been reported in the for the human population, and that's just what we know. There might be so much more out there. So, and we don't have a prediction system for each one of those alleles. Of course, we have the concept of HLA supertype and other things, and we could look at it from that context. But nonetheless, the point here is there's some limitation if you wanted to just do prediction. And of course, some of those predictions might be wrong. So this could be an overestimate, but this is still better than doing a wet lab. So we wanted to use the computational approach, but we couldn't do the prediction approach because of that limitation in terms of computational models available. So then we actually decided to kind of treat the whole sequence as relevant for antigenic diversity, and we made this on two bases. We made this assumption on two bases, that if we are designing vaccine and we want to consider the whole human population, and of course this is in the context of cellular immune response. If you want to consider the whole human population, then we have 25,000 plus HLA molecules, and for a given HLA molecule in a protein, you would have at least 1 to 5% that would be recognized. So if you now consider all the HLA molecules, maybe 100% of the protein would be relevant. If we are looking at the human population level, at individual level, of course, it's going to be much lower, but at human population level, it would probably come pretty close to the whole sequence. So the whole sequence is relevant to somebody in terms of maybe recognition by the HLA molecules of the immune system. So in that context, then we assumed that everything would be relevant for antigenic diversity if we posited at the population level. So therefore, we thought, given that, then we don't need to predict the epitopes. We just study the whole thing, and of course we use a k-mer of a certain length because epitopes are of a certain fixed size in general. So the size that we chose is 9 amino acid. That's the typical size of HLA class 1 epitopes, and it's the core of HLA class 2 epitopes. So we decided to use that size when we study the sequences, and we can use a sliding window, so 1 to 9, 2 to 10, and then we look at the diversity within those k-mers. So moving on, the idea of a minimal set. So I just wanted to quickly explain this. So we talked about studying the whole sequences. So we take a sequence and we study the whole thing in terms of for our antigenic diversity analysis. So here's an alignment just to illustrate the idea of a minimal set. Of course, what we did eventually is not non-alignment based, but this really helps understand what do we mean by a minimal set. So imagine you collected a bunch of sequences and you remove duplicates. So it's a given that you remove duplicates. So you end up with 3 sequences, and these 3 sequences are unique. As you can see here, there's F. They are the same, but there's an L here, and there's a Q here, so that makes this sequence unique. This sequence was already unique from the other 2, but there's also Q, which makes it unique. So the Q's match here, but the L is different. So the second sequence. Well, essentially, you can see from here that all the 3 sequences as a whole are unique from each other. So now what you do next is you generate this so-called k-mer. We chose 9. I've already explained why 9. You could do different k-mer length also. So you create this repertoire of overlapping 9-mers that you can get from these sequences. And this is what we could refer to as the peptidome, or the viral peptidome, or the peptidome of our data set. So you generate this peptidome of a certain k-mer size from each one of those. So these are the k-mers. And then you try to see what is the minimum number of sequences you would need to capture all these k-mers. So you can see from here that all the k-mers here are shared among these 2 sequences, and all the k-mers here are shared among these 2 sequences. And these k-mers are shared across all the 3 of them. So we could immediately see that actually one sequence seems to be redundant in terms of k-mers. So you could just use 2 sequences and capture all the repertoire of, sorry, you could just use 2 sequences to capture all the repertoire of k-mers that were there in the data sets. All right, so this sequence is the redundant one, and you could just use these 2. So 2 out of 3, so that's the reduction you make. You can lose 1 sequence and you wouldn't lose the repertoire. All right, so that's the idea of the minimal set. And this is actually something that we had applied previously about 15 years ago. This was a work that I did under the supervision of Prof. Bruzik, and we wanted to revisit this because this work was applied on dengue, was applied on influenza, but we had not applied this beyond those 2 viruses. And it was quite interesting to us to see how this would actually pan out for a lot of the other viruses out there, especially if we wanted to analyze all viruses known to date, the sequences of all viruses known to date. So we set out mapping the minimal set of the viral peptidome. And by viral peptidome here, I'm referring to all known viral sequences at that point when we did this study. So to break down the objective, we wanted to look back at the algorithm, and the algorithm, we couldn't find the file, it's been that many years. And we had to kind of build from scratch again. And we also realized there were a lot of issues with the algorithm, not in terms of being big data scalable, not in terms of what it could do, but in terms of big data scalability. And it was also not optimal in many ways. So we ended up trying to optimize it and make it big data ready first before we went to do our analysis. And then we also wanted to understand what are the factors that can affect the minimal set of sequences, and then we went out to generate the minimal set. And then I'll briefly talk about how we applied. We went on to investigate how this affected across lineages, viral lineages. So this is a quick methodology overview. So you collect the data. Of course, we went to NCBI, we collected the nucleotide and the protein data. We put the nucleotide aside just for reference, and we went on with the protein data. We removed the duplicates, and then we optimized the tool, and then we investigated the factors, and eventually we did the analysis. And I will go through this. I'm going to skip this. All right, so this is the data collection part. We can skip this. We just explained. All right, so let's focus on the optimization of the algorithm. So this is actually the old algorithm. This was published as part of my thesis then. And I'm not going to go into the details, but essentially the idea is that you have all these sequences, you generate the peptidome, you do this overlapping k-mers, and then you have this k-mers, and of course you remove duplicates, you end up with unique k-mers, and then try to see how many sequences you need to capture all these unique k-mers. So first sequence might capture all the 50% of them. With the second one, cumulatively, you capture 91, and with the third one, you're already capturing 100%. So you only need 3 out of all the many in order to capture these. All right, so we ended up developing 3 algorithms: iter mean, rank mean, and unique mean, because we were just trying to, progressively, explore how to make it better. So the first one was was accurate, but speed-wise was very slow. So it was not big data ready, and we're talking about like 5 million sequences. So we definitely needed speed. And one estimate gave us that we would need 7 months with iter mean if we didn't optimize the speed. So we just couldn't wait for 7 months to see the results. So rank mean, was speedy, but it suffered in terms of accuracy. And then we eventually came up with unique mean that was accurate and also very speedy. These are just some of the limitations of the old algorithm. I'm not going to go through this, but as I mentioned, there were a number of limitations, and we wanted to overcome those. And eventually, unique mean, in some sense, overcame all those limitations, nearly all of those limitations. All right, so I'm going to skip that. So here are some results just to give you a flavor of how they performed. So with 1,000 sequences, that was actually the actual data that was used for Ka-Khan algorithm back then, compared with the new sequences that we use for the other tools. So we don't have the data for this because this was not done back then. But in terms of reduction, there was a reduction of 9.4%, but iter mean, as you can see, was very slow. But was able to reduce at 26. So it was much better in terms of reduction. So there's some optimization that was done, and we could actually achieve much more compression. And rank mean didn't perform very well in terms of the reduction, but actually was quite fast. And unique mean was equally fast, but reduction matched iter mean. So accurate and fast. And we wanted to be sure whether this less than 1 minute was always going to be something like that or close to that. What if the data was quite big? Would we still see a very speedy analysis? Of course, it would still be fast, but how fast would it be? Just to get a sense of that, we actually then tried it with 10,000 sequences. As you can see, iter mean was very slow, and we got a compression of 43%, and rank mean was fast, but there was a problem there. Unique mean was able to do this in 8 minutes, so that was still acceptable. 10,000 sequences, 8 minutes. Of course, with 5 million, you'll probably need about 2 months, which we could, tolerate, but actually ended up being much less. If I'm not wrong, about 18 days, we were able to get the results for the 5 million. Anyway, so this looked good, and we felt that we were ready to then proceed with our next step. So in conclusion, unique mean offers the best combination of speed and optimality to generate the minimal set. And we were able to actually achieve this thing with unique mean because we recognize there's something called singleton monomers that are only found in one sequence. So immediately they become candidates of the minimal set. So what we did was we tried to filter them out, and that really reduced our data set, and then we could do a lot of things much faster. So there was this thing that we recognized, and we separated, and that actually speeded up things. And of course, there was some other tweaking that was done, and some other codes that we used. And of course, multi-core, being able to use multi-core was one of the other reasons for that performance. And with better compute, this should perform much better. And of course, so we eventually, and we have in some sense demonstrated that the tool is applicable to big data. All right, moving on. So we then went on to analyze the factors affecting the minimal set. So these are the list of factors that could affect the generation of the minimal set. The sliding window size, what k-mer you use, and I explain why we need to study in the context of k-mers. Sequence number, how many sequences. So big data, of course, would affect the length of the sequences. Proteins are of different length. Viruses, proteins can be of very different lengths. And the region, like in terms of conservation, if you're studying only conserved sequences, diverse sequences, of course, they would impact the minimum set in a different way. And actually, these three were already studied in the earlier study, except that for the sequence number, the number that was studied was much smaller, and we wanted to revisit that with a much larger data set. The other thing, of course, that was not studied is sliding window. So we wanted to also investigate that. So this time round, we focused on sliding window and sequence number. But what did Khan et al. Observe for sequence length? For sequence length and sequence number, well, in general, if your sequence number increases, you can, well, intuitively you are not able to compress much. Okay, your ability to compress reduces within growing number of sequences. And in terms of sequence length, with growing number, with larger proteins, of course, it was much harder to compress because of larger k-mers that you would generate, and sequence region also it varied. For example, highly diverse regions would be harder to compress compared to highly conserved. So it was all intuitive, but it would be good to capture these in terms of the actual plots. So then we set out to do that, and of course we wanted to make sure that we are correcting for confounders. So when we want to study the k-mer, confounder would be the diversity of the sequences, the number of sequences, the length of the sequences. So we needed to control this. And for diversity, we used Shannon entropy to study that. And this is a formula for Shannon entropy. I'm not going to go into details. You can read our papers. And we have actually applied this on a number of viruses. And the highest we have seen is actually 10 observed for HIV, clade B. Theoretical maximum is 39 that you can get for monomers. And the highest we have observed is 10. So 10 is, so HIV is rather plastic in that it is able to achieve that kind of entropy level. And entropy, basically, the higher the value, the more diverse is the region, the lower the value, the more conserved is the region. So we actually then, given the data sets that we wanted to use, we thought we needed to classify the diversity into 4 areas: conserved, semi-conserved, diverse, and extremely diverse. And based on our understanding of what we have done in the past with previous viruses, we were able to identify those that would fit in each one of these bins. So we further classified this part 1 here into 4 bins so that we could understand the minimal set with respect to each one of these levels of conservation. And so these were the data sets that we used to do our factor analysis. You can see the entropy value would correlate with the grouping that I mentioned earlier: conserved, semi-conserved, diverse, extremely diverse. And for the sliding, when we did the sliding window size, we explored 3 mers to 23 mers. This is the range that we felt was relevant for immune system, and not just, of course, for cellular, it doesn't go to 3, but I think in terms of antibody epitopes, some of them can be much smaller, but we just, for curiosity's sake, we just went from 3, which is where I think most people say is non-random, begins to be non-random. BLAST uses that 3 amino acids. So we went from 3 to maximum about 23. That's the range I think class 2 can recognize. So we took that whole range, and when we did that, we fixed the number of sequences and we fixed the length of sequences. So number was fixed to 100, the length was fixed to 100, and then we of course went diversity by diversity, different diversity levels, and we went across the different k-mers. For the number of sequences, we analyzed from 100 to 1000. Again, this is not really big data, but it was much larger than what we had done, Kanetal had done previously. And of course, when you do this, you fix the length and you fix the window size, and we chose 9, and which the reason I've explained before. So this is the result now, factor sliding window size. So in general, what we could say is that the reduction, antigenic reduction, basically how much could you compress? So the antigenic reduction was the highest for the conserved, and I think that's intuitive. Conserved, there's a lot of k-mers that would be redundant, so you could really compress them. And then semi-conserved next, diverse next, and extremely diverse was the lowest in terms of compression. But we also noticed that, as your overlapping k-mer size increased, the ability to compress also reduced. It was more or less flat for conserve. It didn't change much. Similarly for semi-conserve, for diverse, it started also flattening out at around 23. And similarly for extremely diverse. So there was this, you were able to reduce further, and then it just started to flat out. All right, so that was the observation for sliding window. And in terms of sequence number, as I mentioned earlier, Kanetal had studied this, and if you increase your sequences, your ability to compress just reduces. But nonetheless, of course, you can compress, conserve, and semi-conserve a lot, followed by diverse and extremely diverse at a small number of sequences. But as the number of sequences increase, your diversity, well, the pool of variation, because these are unique sequences, right? So there would be natural diversity increment there. So then it starts tapering off, starts flattening out in terms of reduction for the semi-conserve and diverse. And we actually were quite curious to see how this would actually go for the extremely diverse, and we observed later on. So this was a unique observation that extremely diverse actually, can continue to go up to the point that we have seen in some cases it actually beats the compression you achieve in terms of percentage with the conserve and semi-conserve. Right, so there's a bit of we should actually redo this with much larger data set, and there's a twist here that extremely diverse could actually overtake diverse and semi-conserve in terms of how much you can compress. All right, so we will, bearing that in mind, proceed. So in conclusion, for this part, antigenic reduction was inversely correlated with increase in Kmer size. So you increase the Kmer size, your AR reduction, the ability to reduce slowed down, you flattened out. Antigenic reduction was directly correlated with increase in number of sequences. So as you increase the number, your antigenic reduction also increased. And AR was the highest for the conserve and the lowest for extremely diverse data set. But as I mentioned earlier, this actually flips for some viruses with a lot of data and a lot of diversity. So moving forward, generation of the minimal set for all known viral pair sequences. So this is when we really set out to do what we wanted to do from the beginning and apply this on all viral sequences, all those close to 5 million back then. So we took all our sequences. First, of course, we removed the duplicates. So it was nice. It was interesting to see that half of the sequences were duplicates. So that's an observation. And then we were left with 2.1 million, and we were able to compress this further, not so much actually, only about to the tune of 16%. That was the antigenic reduction through unique names. So we ended up with 1.3 million sequences. And now as a whole, I think if you sum up together the duplicate reduction and the antigenic reduction, that's a total reduction of 73.4%, which means we are only left with 26.6% sequences of the input sequences that would be relevant in terms of antigenic diversity. So that's a huge reduction, and there are a lot of implications of this, which is, that if you wanted to study antigenicity and all that, you could be just focusing on this 1.3 million sequences, especially for those people who don't have a lot of compute. This would be much more manageable data size. And also if you wanted to do wet lab experiments, this is definitely much more manageable number of sequences to be studied. Well, still big, but much smaller than 4.9 million sequences. So there was this interesting observation that how much we could reduce in terms of totality, but antigenic reduction itself was actually not so much. So the 56% reduction, the high reduction up there implies that there's a lot of maybe sampling bias, there's a lot of duplication that's going out there. And the antigenic reduction being rather low indicates that there is still a lot of inherent diversity within the sequences, and therefore that we were not able to compress much there. So in conclusion for this one, the high duplicate reduction reflects possible sampling bias in the collection and reporting of viral sequences in public databases, and this is there, we have seen it, usually sequencing would be not spread out. People would be going for the same sample sites and maybe neighboring sample sites. So there's that tendency to pick sequences from a certain sample sites rather than be more widespread, more uniform. And relatively low AR observed for the viral peptidome suggested a high diversity of the viral superkingdom. So the viral superkingdom, it's actually very diverse in general. And we can, of course, we can relate to that with the fact that there's so many different species of viruses out there. And the AR, we predicted that this AR will range from 15% to 25% with over time with further increase in data. The reason for that is you already have all this diversity captured. New sequences that are coming in are not going to drastically increase that diversity. I mean, in terms of, let's say, Kmers, you're not going to get a lot of new Kmers. Even if a new sequence comes, majority of the Kmers will be exactly the same as the previous sequence. Maybe there's one mutation here and there. And those mutations will result in some new Kmers, but the majority of the Kmers are going to be more or less the same with the existing data. So in terms of the AR compression, we think that this is, well, 5 million is quite a big data. So we think that this number is not going to change much. Will go up and down. Why would it go up? Because, well, if this existing data is not really representative, we end up sequencing some novel viruses, then there would be some changes. But if you're not going to sequence something novel out there, and this is reflective of what's out there, then of course it's not going to change so much. But nonetheless, this is what we think it would be, and of course it'll be interesting to do this maybe few years later, just to see what's the status then. And the last part, actually, I'm not going to go into detail because of a time issue. But once we had built the tool, we applied it on all viruses, and we saw what it looked like. Actually, the next thing we wanted to do is to do this across species, genus, family. So like take one sequence, take one species, and then take the genus of that species, then take the family of that species, and try to chart out this landscape of minimal sets across this lineage. And so we have actually a lot of results there that I don't have the time to go through, and hopefully that will be another talk someday, and we'll be able to share. But also all this work is currently being prepared for publication, so you hopefully will be able to read these results in the near future. So that's really one nice application. Now, as I mentioned, the issues with alignment-based approach, you could mostly do it at the species level. Going beyond, it's going to be very challenging. So with this tool, we can now study the diversity at a species or a genus, at a family. We could even go where we have already gone to the root, which is all the super kingdom, which is all viruses, and we've already shown the results. But we wanted to now zoom into the various lineages and see how it changed across lineages. All right, so in the interest of time, I'm not able to go into those results. I'm just going to conclude now. I think I'm more or less hitting the time limit. So our tool, Unique Mean, which is publicly available, enables analysis of a minimal set for a given sequence data set of interest. And as I mentioned earlier, this is really an enabler because it allows us then to go beyond a particular species, to go beyond alignment, and to be able to study any viral sequence that we are interested in. And of course, actually, the tool would work with other organisms, for example, bacteria, if you wanted to. We have not tried this, but we think it would also work with bacteria and others. Actually, we had a project to do the same thing with bacteria, but bacteria we have so much more data, 700 million sequences. So for that, we really needed much more compute power. So that's something ongoing, and hopefully we'll have some results in the future for that. The project is a little on hold because of that compute, and hopefully we'll be able to share something there. So the tool is an enabler. I'm not saying it's perfect tool. It probably needs more improvements, and hopefully we can improve this and make it much more robust in the years to come. Also, I think the second key point is Unique Mean is able to reduce big data to a reasonable working size, and there's a lot of value in that in terms of kind of zooming in and helping you just focus on the sequences that you should be focusing on when you want to study antigenic diversity. I think it has some positive implications to wet lab validation. You don't have to be overwhelmed with all the data that's out there. You could focus on much smaller data without worrying about losing the possible repertoire of k-mers that would be immunologically relevant. And also this is very useful for those people who don't have the compute. Not all of us are blessed with a lot of compute power to do this kind of analysis. So this can also be helpful in terms of shrinking the data size so that you could focus on a much more manageable data set. And lastly, the low AR antigenic reduction that we saw when we did all viruses observed for all of the viral peptidome suggested that the viral superkingdom is actually inherently quite diverse. If this is what we have seen, then it's going to hover around that. But if this is not everything that we have seen, and there might be a lot of novel viruses that are yet to be sequenced, which I believe that would actually more likely be the case. I don't think we have sequenced everything, so there would be some novel. For example, Mimi viruses was a novel discovery a few years ago. Giant viruses. I'm sure that if we do these fishing expeditions, we might discover a lot more novel viruses. So, but nonetheless. we estimate that this is going to range, the AR is going to range from 15% to 25%. So you at least get some rough idea what kind of reduction you can get. But for the DR, duplicate reduction, it's very hard to say what we will get because it really depends on the sampling and it varies from virus to virus. So that's something, it's a bit hard. For example, you could get a duplicate reduction of 90%. You just reduce by 90%, and this we observe with Ebola virus. And for some viruses, maybe you get much lesser. So actually, this is something else we wanted to go back and look, do a factor analysis on duplicate reduction and see what we can learn from there. So that's all I got to share with all of you. This is the GitHub page of the student Chong that was made by the student Ms. Chong, and the tools are available. So if you're interested in applying this, we're happy to collaborate and work with you. And I just want to end with acknowledgement. Again, a big thank you to Chong, whose photo is here. She did all the hard work here, and credit goes to her and bringing the project to fruition. And she has completed her master's. And I want to acknowledge Dr. Kenneth Ban of National University of Singapore for giving us some compute power to run these to run the tool. Dr. Choi Sui-Bing, my colleague from Perdana University, for assisting and co-supervising the project, and Mr. Lim Wei Lun Liner for working with Chong and doing some of those code tweakings in optimizing the code. And last but not least, Vladimir Bruzik, my former PhD supervisor, who kind of initiated this work about 15 years ago. And we also received these fundings to support the project. Thank you very much. And I'm happy to take any questions. I hope I didn't exceed the time. I have kind of lost in terms of time. So I hope I'm still okay, or my apologies if I exceeded.
All right, sir. That was really a great talk. And now I would like to ask the participants whether you have any questions. We are now open for questions for 1 or 2 minutes.
Akila, shall I ask now?
Yes, sir. Please go ahead.
So, Dr. Asif Khan, sir, it is a wonderful talk on viral peptidome and antigenic reduction. So, in this, there is 1 or 2 queries are there which I would like to get clarified from you, sir, Asif Khan.
Sure.
So, what nowadays mRNA vaccines are coming, will they be promising, or do they replace our antigenic vaccines? If they replace, what is the procedure, how to trap mRNAs in a large scale? What we are doing for the protein collecting, trapping in a large scale is convenient rather than mRNAs collecting and trapping in a large scale, ampule.
Right. Thank you for that question. This is definitely very relevant and pertinent with mRNA vaccine approaches being used, to combat corona. So in terms of the antigenic diversity, I think at the end of the day, whether you use mRNA or DNA, that's more of a delivery mechanism. At the end of the day, the immune system recognizes peptides. So the actual recognition happens at the peptide level. mRNA, DNA vaccine approaches, which were there before that, facilitate the delivery because peptides are not so stable. So when you infect with a peptide, they may get degenerated easily, and there are a number of issues with that. So I think these approaches are taking care of the delivery mechanism. Eventually, what the immune system recognizes, I have not read much of this yet, so I might be off, but what I know from the immune system is that it will recognize a peptide. So at the end of the day, you still need to deal with peptides, protein sequences, and the mRNA is going to code for a protein, which will hopefully will get chopped accordingly and that be presented to the immune system. So this is still relevant, and this is still, it kind of doesn't change in terms of actual recognition by the immune system. Even antibodies, I don't think antibodies are going to recognize the mRNA, but rather it's the protein that's coming up. I think this is still relevant.
With regard to the delivery mechanism, there must be some, what is it called, techniques that we have to develop, particularly for mRNAs.
Yes, yes, and I think this is a novel area, and
Novel area.
I see a lot of promise there.
Yeah.
And
Another important
developments.
feature, Asif Khan sir, another important feature, you were mentioning about the conserved regions and antigenic reduction, all those things. What I wish to let you know is, if there are conserved regions among 5 or 6 pathogens, if we prepare a vaccine taking into consideration the conserved regions, will not this conserved region will fight against those 5 or 6 pathogens for which we designed the vaccine?
Yes, yes. Actually, this is the premise behind the search for universal vaccine against influenza. Influenza, for example. So
H1N1.
Yes, influenza. So there are a lot of papers that talk about universal vaccine, and the idea is if you find something conserved that's across the board, then it would work. The problem is with influenza. Actually, my colleague Haney, a long time ago, for 13 years ago, actually published this paper on H5N1. When you go look for something that's conserved across different subgroups of the virus, for example, different subtypes of influenza, you do see something that's conserved, but the number of these sequences that are conserved across is much smaller. You don't have that many of them, and even if you do, they're very short. So then you don't have a lot of sequences to play with in terms of trying to see if they're really optimal in terms of immune recognition. So that's one challenge out there. Of course, the alternative, and in dengue also we did an analysis and we proposed something called pan-dengue virus sequences, sequences that are conserved across each one of the dengue. And the idea was that this might mitigate the original antigenic sin because it's conserved across the 4. And we found 44 sequences that were conserved across the 4, but they were not 100% conserved. So there were still some small variants within each of the serotypes. But we found 44 that were shared at 80% or more conservation. And there were like 6 only that were 100% conserved. So your number shrinks when you're looking at higher conservation. And if these 6 turn out to be not immunogenic or poorly immunogenic, then you're left with nothing. So that's the challenge with the universal vaccine, but definitely that's the idea if we could. So the alternative to this so-called pan or universal vaccine is to avoid original antigenic sin, and we have just published a paper on this, which is to look for like species specific and highly conserved. So you could find something that's maybe in the case of influenza, let's say H5N1, highly conserved in H5N1 and species specific to H5N1, and then find something that's highly conserved in H7N9 and specific to H7N9. So there's no cross-reactivity, because cross-reactivity is something that we don't fully understand, and this is where original antigenic sin can happen. So for dengue, we proposed, you find like serotype 1 very conserved, but different from serotype 2, and different from serotype 3, different from serotype 4. So when you get recognition, immunity, memory response to that, this memory cell will not get confused in recognizing the other serotypes, and for the other serotypes, you will call the other response. So there's a lot of work going on in this, and both ideas are interesting, and hopefully something comes out of that.
Another interest, conserved regions need not be RBD, receptor binding domains.
Yes, it did not be. For example, in the case of cellular immune response, the protein gets chopped, and then and so you have the availability of these chopped peptides that will then bind to the HLA. And the HLA doesn't prefer something that's from the RBD. It could be any part of that protein. So for cellular immune response, it's RBD. It doesn't matter.
So one more important point, Khan sir. Your Malaysian people HLA and Indian people HLA, if you make the vaccine, will it differ?
Well, in Malaysia, we do have Indians there, so maybe there will be some commonality there, but you also have the Malay population, you also have the Chinese population, and there will be diversity given that. And also in India, you have a huge diversity in India itself. I mean, India is so big and so diverse, so you would actually struggle to find commonality among Indians itself, if you consider the whole population.
Sir, the last point is the SARS-CoV-2. If you prepare a vaccine, will it work against MERS and the SARS-CoV-1 also?
Okay, that's a very good question, and I was very surprised to see that the MERS sequences are actually very different from the current SARS. If I remember correctly, they were like only about 20 to 30% similar. So there's a lot of diversity. So most likely they won't work because these coronaviruses are quite different from each other.
Yeah. Thank you, sir. Whenever you come to India, please tell us. We'll host you for some time.
Thank you. My pleasure. I was happy to be part of this. Thank you.
Yeah, we've got good friends from Malaysia. They came to our place and spent here for a week time.
Nice, and likewise, if you come to Malaysia, do let us know.
So with the permission of the chair, I just have a small question to Asif. Yeah, please. Yeah, Asif, it's a wonderful it's a wonderful talk, Asif. Thank you so much. So I was just wondering, how minimal is a minimal set? Can we just draw to a conclusion that they must be or they could be this much minimal? Just like in your case, you have drawn to a conclusion that you have reduced to 44%. But what if if a future pandemic would probably come up with a new viral peptidome data sets? What would be the minimal for a minimal data set?
Well, the minimal for the minimal data set should be whatever is in nature. I think nature would know the answer to that, and we looked at the available data that has been reported from nature, and what we got was for all viruses, the minimal data set was 26%. So you could compress it by 73%, and you're only left with 26% that would represent. So that's what nature told us based on what we have. And as we see more of these viruses, and if we discover novel viruses, then we will get closer and closer to the truth, the actual truth number. So I mean, overall nature, we don't have all the data. So that would be the truth. And what we observe is a reflection of that truth. So we are right now standing at 26%. And with more sequencing, we will get closer and closer. And we suspect the actual number, as I said, hovering between 15 to 25%. So the final, let's say if we could sequence everything available in nature, then we will know the actual truth value, but we are not there. But we suspect that actual truth value would be somewhere between 15% to 25%. So we don't have any particular desire that it should be here or it should be there. It's really what it is that nature would elucidate to us eventually as we sequence more of these. But we'll probably never get there. Yeah.
Thank you for an excellent talk. Yeah.
Thank you, Prash.
Thank you very much, sir. I feel this is a technical quiz. And now, due to time constraints, we'd like to move forward for the next speaker, Dr. Tikam Chand Dakal, Assistant Professor, Department of Biotechnology, Mohanlal ML, Sukhadia University, Rajasthan. Sir, please pardon to ask you, we would like to ask you to conclude the session within 25 minutes. I hope you take this on a positive note and continue the session. Thank you very much, sir.
Dr. Tikam Chand Dakal is currently working as an assistant professor in the Genome and Computational Biology Lab of Department of Biotechnology, Mohanlal Sukhadia University, Udaipur. Dr. Dakal has more than 12 years of research and teaching experience. Dr. Dakal pursued his PhD studies from University of Modena and Reggio Emilia, Italy, and has postdoctoral experience from University of Montreal, Canada, and University of Bordeaux, France. Dr. Dakal has 40 publications in peer-reviewed journals of international repute. Dr. Dakal has been conferred with the prestigious FIMS Young Scientist Award by FIMS Society of Germany in 2013. Dr. Dakal is on the editorial board of several reputed journals, such as Frontiers in Microbiology, Frontiers in Genetics, etc. Dr. Dakal also holds research chair in BioClues organization currently. He is engaged in elucidation of cellular and molecular mechanisms of diseases such as cancer, neurodegenerative diseases, diabetes, and cardiovascular disorders. Now I call upon Dr. Tikam Chand Dakal to take upon the session. Sir, please.
Am I audible to all?
Yes,
Yes,
sir,
sir.
you're audible.
Okay, okay, fine. And my screen is also visible to all?
Yes,
Yes, sir,
sir.
it is.
Okay, okay. So first of all, I would like to thanks Professor Rangarao, Professor P.B. Kavi, and the whole organizing team for giving me this opportunity. And I'm also thankful to the two students or the two faculty members there who have really introduced me very well. And so you already took 5 minutes of mine. So now I have only 20 minutes to cover and finish all my talk. So thank you once again. So without wasting time, I would like to start my presentation. And today I would be presenting my work. Basically, it's a bioinformatic analysis of SARS-CoV-2 spike protein, step towards designing COVID-19 therapeutics. Basically, before going into the presentation, I would like to just ask, there were many questions, like when the lockdown started, since that time, I had some questions in my mind that why this coronavirus? This is not a novel virus. This is not a novel virus because from 1960s or maybe from 1930s, we have such kind of viruses and humanity has already affected from these kind of viruses. So it's not a novel virus, but basically when we say it's a novel virus, we say it because this virus is completely different from the previous coronaviruses. We had the MERS, we had the SARS-CoV-1 earlier, but this is completely a different kind of virus, and the reason of this that this virus is very different is that it has a very high transmission rate and it's very highly infective also. It's very infective. So this was the question in my mind, and it, of course, in many of you also have that this question in your mind, that why this coronavirus is highly infective. And second question is that in COVID patients we often see complex symptoms in sense that they had the high fever, as if they are infected with dengue or malaria. They vomit, they have nausea, as if they are infected with salmonella or some food poisoning or something like that. They have a breathing problem. They have like breathing problems as if they are asthmatic, or they have some immune disorders. Like so, there are so many complex symptoms that we can see in the COVID patients. But until now, I could not find that why we have this type of complex symptoms in COVID patients, and why the pathophysiology of COVID-19 or SARS-CoV-2 infection is highly complex. Then the third thing comes like the vaccine. So is a vaccine like is now like a long waiting thing for all of us. We all are waiting for the COVID-19 vaccine. That the vaccine could be of whole organism vaccine. Also, it could be a recombinant vaccine. It could be the mRNA vaccine, which Professor he just asked a question to Asif right now, a few minutes ago. This is also a question in the mind that what would be the nature of the vaccine, and how we would be able to restrict the infection of the COVID-19. So this is also the question. Another question is that what could be the novel therapeutics? Like when we talk about the vaccines, it's really a very expensive affair. So I do not know, because now so far we do not have a vaccine in hand, so we cannot talk about the cost. But I guess the cost would be like very high, and it would not be like affordable to all. So and the another thing is that uh, like once I had a talk with Professor Chaitanya from GBRC, and he had something in his mind we people many in India also, like we are sequencing the virus, and we are getting to know about okay, this virus has this kind of mutations, and the virus has mutated so far. It has mutated already 100 times. So because this is the knowledge about the problem, this is the information about the problem. So but we have to think about the solution of the problem, and the solution of the problem is that we have to understand, we have to know that why some people are getting they are more prone to this virus, some are less prone to this virus, some are susceptible, some are not susceptible, some are even not getting infected this virus, and some are easily getting infected with this virus. So what is inside this the genome of the human. We have basically to do a comparative genome analysis of the COVID patients and the patients or the people that have that are that are not getting infected to this infection, even if like in India we do not have such like a very strict lockdown situation so far. So we are going everywhere, we are touching everywhere. So it means that we are exposed to virus. But what makes us to be resistant to this kind of virus? So we have to do some comparative type of analysis, comparative genome analysis, proteome analysis, or some kind of analysis to find out the reasons that what is inside our genome that is making us prone to coronavirus, and what is inside our genome that is protecting us from the coronavirus. Of course, we have some knowledge, and but we have to think about in this direction. So I will so I will skip some of my slides, because I already have given the introduction. So when we talk about the coronavirus, like it's like a hell coronavirus. Like students, they cannot go outside, we cannot go outside in the streets. It was a situation earlier. People are like marrying. They cannot marry because now there are so many restrictions also. But of course, some people are taking advantage that they can marry at very low price also. Everything we have to do online. Of course, online things are very good. Earlier also we were doing the things online, but like taking the classes online and doing everything online is little bit difficult task also. And the situation of a class where teacher is taking the class and students, they could be sleeping, and the situation is very drastic. So we just have to stay at home, and we have to just stay protected from this coronavirus. This is the part which I would really like to skip, because I have already and I think we all know that the family of the coronavirus and the lineage of the coronavirus. And this is the natural history of the coronavirus. That okay, this as I just mentioned, that the coronavirus is not a novel coronavirus, it's not a new coronavirus. Earlier also, since 1930, we are getting infected. Like people or the mankind, we are already they had this kind of virus on Earth. So I just skipping this one also. So here, just I would like to mention that we had the MERS coronavirus, the SARS coronavirus. Sorry for this this spelling mistake here. There is some typos here. This is SARS-CoV-1 and then SARS-CoV-2. So we know the hosts of these coronavirus, but we do not know the intermediate of this. Especially in the case of SARS-CoV-2. So this is the outbreak of the SARS-CoV-2. This is the pandemic situation we have, and this is the situation. Like if we just compare this the SARS infection of the SARS-CoV-2 and the SARS-CoV-1, and we can imagine that the SARS-CoV-2 in 8 months, there were only 26 countries affected from this type of virus. But now we have like more than 250 countries that are already affected by this virus. So I will just skip this part. This is the genome of the SARS coronavirus. We all know about this genome of the SARS coronavirus. I'm coming directly to this the spike protein of the SARS coronavirus. Basically, this is the spike protein of the SARS coronavirus that attaches to the host cells, and it's give entry to the viral genetic material to get inside the host cells. So it's basically the spike protein. So it is the and also the spike protein is the protein that interact with the host cells. So it is the first in if we talk about if we talk about the first interaction of the host cells with the virus, it is because of the spike protein. So So that's why many of these studies, the majority of these studies are focused on spike protein of the SARS-CoV-2. So this is how coronavirus multiplies inside the host cells. This is from one of my paper which has been accepted already. This is the multiplication of virus, the same thing. This is basically the immune response to SARS-CoV-2 that it's the normal picture where I like to present. I like to show that how the SARS coronavirus 2 when it would like target our cells, what kind of immune response and how the immune response will be mounted against the SARS-CoV-2. So I would like to start my bioinformatics part. What I did on SARS-CoV-2 is basically that we took the SARS-CoV-2 spike glycoprotein sequence, and we tried to find out the antigenic regions on the spike protein of the SARS-CoV-2. So if we would know the antigenic regions in this spike glycoprotein of the SARS-CoV-2, we would be able to know that what is inside that those antigenic regions, and because of that we have the complex symptoms in case of COVID patients, and some other things that we will come across in a few minutes. So basically what I did here is that I took this SARS-CoV-2 protein sequence and tried to find out the antigenic sites on the spike protein, and also tried to see the CTL epitopes against the MHC class 1 and MHC class 2 that are on the SARS-CoV-2 spike protein, basically. So these surface exposed antigenic sites here, you can see here. What you can see here is basically the antigenic sites that would tell us that why we have the complex symptoms in case of SARS-CoV-2, and the CTL epitopes and CTL epitopes in SARS-CoV-2 glycoprotein, spike glycoprotein would tell us that which peptides or what are the peptides that we can take for the vaccine development, basically. So this is what we found in our study, that we found almost like 9 to 10 antigenic epitope sites of the SARS-CoV-2, and these all antigenic epitope sites were like they were all surface exposed. It means that they were not like residing inside the spike protein. They were on the surface of the spike protein. And it means that they can interact with the host cells. And if we consider them as this antigenic one, so maybe these antigenic regions, these antigenic sites would have something. That's something I would tell you in a few minutes. That have something that is basically mounting the immune response in the same way that we have basically in case of salmonella infection that we have sometimes in case of malaria or dengue. So basically what I did when we got this antigenic sites, when we predicted this antigenic site, we used a tool. We use a tool and we try to see that if whether any of the microorganisms or any of the parasites, they have this the molecular similarity with this antigenic site that are present on this spike glycoprotein. So to our surprise, we came to know that there are 54 antigenic determinants, and we found just in this spike protein of this SARS-CoV-2. So there are 54 antigenic determinants that we found and were related with the 12 pathogenic bacterial species like Mycobacterium tuberculosis, Mycobacterium leprae, Anthracis, then Clostridium also. We had the Clostridium tetani also, Helicobacter, Listeria, Staphylococcus, Streptococcus, Vibrio, and Yersinia pestis also. Then we also found the similarity with two malarial parasites, means the sequence from the two malarial parasites and influenza virus also. Basically here what I like to say is that this is basically what we found here, that on the top we have the antigenic sites which we predicted in this SARS-CoV-2, and on the x-axis we have the microorganisms, and if the box is colored, it means that that particular antigenic site has the molecular similarity with the Mycobacterium tuberculosis, Mycobacterium leprae, like that. So this was the first study. I haven't seen any study until now so far that could basically explain that why we have the complex pathophysiology in case of SARS-CoV-2. It is basically because the antigenic sites that are present on the SARS spike protein of the SARS-CoV-2, they have the sequence, and that sequences basically have the molecular similarity with the proteins that are present in Mycobacterium tuberculosis, Mycobacterium leprae, Bacillus, Borrelia, Clostridium, Helicobacter, Streptococcus, Staphylococcus, Vibrio, and like that. So when a spike protein will have the sequence or the molecular similarity with the pathogenic sequences that are present in pathogens, so it is quite normal that we may have the symptoms similar to those particular diseases also. So that's why this is basically I feel this paper is under review now, and like it is like already I have sent the revision also, and there was like some very hard questions related with the immunology. I have already covered everything. So this is what basically I feel, that is the antigenic sites that are present in the spike proteins, they have the molecular similarity with the proteins that are present in Mycobacterium tuberculosis, and there are other pathogenic organisms. So I'm just talking about the one spike protein for now. So if I talk about the M protein and if I talk about the E protein, the story would be completely different. I mean to say that I tried with some other proteins also, and I found these molecular similarities is there also. So basically this is the same thing, that the antigens from Mycobacterium that showed similarity were mainly involved in modulating host immune response and ensuring persistence of and the survival of the pathogen in the host cells. There were like the antigenic sequences that were present in the pathogens were basically related with the toxins that basically they secrete or they release when they enter inside the human cells. So so it means that the SARS-CoV-2, the sequence of the SARS-CoV-2 has the sequences or the motifs that basically that have the molecular similarity with the another pathogens, and these motifs or these peptides, they basically are mounting the immune response in the same ways as those pathogens do in our body. So this is one my study, and that I also did some study towards designing a therapy for COVID-19 patients, and this work is already published now in Immunobiology. It's a very good journal of the Elsevier. This therapy is basically pulmonary ETTA chelation therapy. So I would just like to skip this part. We all know that all the vaccines, all the neutralizing antibodies, and all the therapies that were designed previously for the treatment of the SARS-CoV-1 were found ineffective for the SARS-CoV-2, and there are so many reasons for that also, but I do not want to go more into the details. And that was basically where we felt the need of developing new therapies for the SARS-CoV-2. So until now, we do not have any drug, vaccine, or any medicine for the treatment of SARS-CoV-2. So if we talk about the like drug discovery or drug repurposing, like we need years, and years to develop a medicine for the SARS-CoV-2. Okay, I'm just like to skip this part and coming to my analysis part. So this is basically the mechanism of virus entry into the host cells, that the spike protein, the virus, it attaches to the host cell membrane via its spike proteins, and then the virus gets its entry. It's not the virus basically, but it's the genetic material of the virus. It gets its entry into the host cells. So what I did in this one is that I took the protein sequence of the spike protein from the SARS coronavirus and the protein sequence of the spike protein from the SARS-CoV-1 one, and I tried to see why there are differences, and what are the differences that are making the SARS-CoV-2 highly infective. So what I found here is that this SARS coronavirus 2 has a very important functional motif that is called as basically the RGD motif. I already had discussed about this RGD motif in many of my talks earlier, and this RGD motif is really a very function. It's a tripeptide motif, but it's a very important, very potent. It's a very strong motif, and if any protein will have this motif, that protein would have a very high ability to bind to the host cells. This is basically what I found. So if the SARS coronavirus, the spike protein of the SARS coronavirus 2 has this motif, so this motif will make this SARS coronavirus 2 to bind to the host cells very effectively and very tightly. This is basically the biology. So what is the importance of this RGD motif? This RGD motif was originally found in fibronectin. It's basically an extracellular matrix proteins having role in cell attachment and cell adhesion. So basically, this motif is present in fibronectin. This is also present in vitronectin and laminin. And all our tissue culture flasks and petri dishes, they have coating of these extracellular matrix proteins, and our cells, our human cells, they attach to the bottom of the plates just because of this RGD motif. So here it is all about the basic science. There is no like a very big science. There is no like a big. There is no like something like I used a very big instrument and I did a huge analysis. But it's really very important to note here is that this RGD motif is present only in the SARS-CoV-2. It's not present in SARS coronavirus 1 or any other previous SARS coronavirus. And because of this RGD motif, this virus can have like a very high potential to bind to the host cells. You know, if we are talking about the lungs, it is able to bind to the lung cells very tightly or very effectively just because of this RGD motif. And this I can say this now because this paper is already accepted now. So it is also important to know that what are the proteins. This spike proteins not going. The virus is not going directly and binding to the lung epithelial cells. Basically, it is binding to the lung epithelial cells through some receptor proteins. So what are the receptor proteins that are basically present in the lung cells, that are basically expressed in the lung cells? So what I did, I tried to see the gene expression data of the lung cells, and I found that this RGD motif can bind to the integrins in the lung cells, and integrins, they have good expression in the lung cells. And the RGD motifs, basically they have a high affinity for the integrins, and when integrins, they have high expression in the lung cells, so it makes some story that yes, of course, this could be the integrins. With the help of these integrins, this virus spike proteins is able to tightly bound to the lung cells, and it's like, no, it is able to become highly infective as compared to other SARS coronavirus earlier SARS coronaviruses. So earlier SARS coronavirus, when they did not have this RGD motif, they were not able to bind to the lung cells very effectively, and so and for this reason the infections were very less. The infectivity was very less. So these are some of the integrins like alpha V beta 6 and then alpha 5 beta 1, and there were some other integrins also, alpha 8 beta 1 and ICAM 1. So these were the integrins that have a strong affinity for the RGT motif, and this RGT motif is present in the SARS-CoV-2, the spike protein of the SARS-CoV-2, and the integrins they have a high expression in the lung cells. So it makes some sense that yes, this could be the integrins, because until now so far we are just talking about the ACE2 receptors, but tell me why the ACE2, why should we believe on the ACE2 receptors? Because the SARS coronavirus 1 was for the SARS coronavirus 1 also the receptor was the ACE1, my infection was less. For the SARS-CoV-2 the receptor is ACE, but why the infection is very high? It means that of course ACE2 receptors could be the receptors, but yes, for me, and now I can say with high confidence that there is an alternate receptor also for the SARS-CoV-2, and the integrins could be one of the very important receptors for the SARS-CoV-2. This is basically it is based on the RGT motif that is basically present in the spike protein of the SARS-CoV-2. So basically, after that, I did some molecular docking experiment also, and where what I did, I took the integrins and I took the RBD domain of the spike protein, and I tried to dock the spike protein onto the surface of the integrins. And what I found there is that the spike proteins is able to like interact with the integrins, and there was something interesting here that the RGD motif that I found is basically interacting with the integrins, and where this RGD is interacting with the integrins on that particular site on the integrins, around that site around the docking site on the integrins, there were the sites for calcium binding also. And this is also a well-known fact that the RGD motif that is present in extracellular matrix protein also, they utilize these calcium and magnesium ions to make this strong bonding even more stronger. So when we use when we use PBS buffer without without calcium and magnesium and with EDTA, we can disrupt this binding of the extracellular matrix protein with the host cells. So same thing is happening here also. So we have the spike protein with the RGD motif. With this RGD motif, it is binding to the integrins. On the integrins, at that particular docking site, around that docking site, we have the calcium binding sites present here. So it is making sense that this RGD motif is taking use of the calcium ions for making its making its bonding with the integrins even more stronger, and this could be the reason for the high infectivity of this SARS-CoV-2. So of course we have to do so many of the molecular experiments also to validate this kind of study. But of course, with the things that are already known about the RGD and that the things that are already known about the integrins, this hypothesis, my hypothesis, was making sense to the editor and to the reviewer, and they accepted my paper also. So that is the same thing where I showed on the right side of your screen. You can see that I have made a a red circle. So this is basically the red circle where this RGD motif is binding to the integrins, and this red circle is basically the calcium binding sites present on the integrins that have the role in facilitating this virus attachment with the host cells integrins. So now when it comes about the therapy, so my hypothesis was that if we can remove these calcium ions from the lung cells, we can we can manage the situation of coronavirus to some extent, So this was my hypothesis. So, so as we do in our laboratory also, that we use EDTA that chelates these bivalent ions, and in a cell culture lab also, this type of techniques is already in in use for so many years, that when we use EDTA, it disrupts the attachment that the cells they have with the bottom of the plates, that the cells they have the attachment with the extracellular matrix protein. So the same thing I try to do here, that if we will use the EDTA, this EDTA will create this calcium ion from the lung cells, and when these calcium and magnesium ions would not be available to the SARS-CoV-2, its chances of like attaching to the host cells through RGD or integrin binding would be very, very
sir.
So yes.
I'd like to remind you the time. Due to time
no
constraints, we would like to ask you to stick to the given time.
Oh, sure. So I'm just finishing in 5 minutes because I'm almost at the end of my presentation now. So so what I thought that, okay, this we can have a therapy with EDTA. So this was the therapy that I proposed, that the EDTA chelation therapy. We can use this therapy for the treatment of COVID-19 patients. And this treatment of this using this EDTA chelation therapy is already a FDA approved therapy. So we do not have to we do not have to get into so many of documentation work to get a therapy approved for the to be used on the COVID patients. So we can directly use this therapy on the COVID patients. So this was basically the nebulizer and inhaler based EDTA chelation therapy for the treatment of COVID-19 patients. So here what we will be doing is that we can have this EDTA. We can inhale this EDTA, and this inhaled sodium EDTA, it would go inside the lungs, and where it will chelate these calcium ions present there, and this calcium ion that is important for the virus and host cells attachment. So in that case, when there will be no calcium ions or the magnesium ions, so the infection or the attachment would be affected, and second, the infection would be very, very less. So this was basically the therapy. So this therapy, I propose this therapy, and this is in Hindi language basically. So there are some additional benefits of this therapy also, because now that we have the time constraint, so I do not want to go more into these details now. So yeah, I just like to skip everything. I had made the presentation like a very big presentation. So this was already in the news also, and I got some award from the MHRD also for this therapy. So now this paper is also out. So finally, I would like to acknowledge Mohanlal Sukhade University for providing me the infrastructure, and of course I didn't put the photographs of my students. So I know they are watching me. So I would like to thank all my students also. They are like very helping to me. Whenever I need their help, they are always ready. And I'm thankful to Professor Pankaj Goel, Dr. Abhishek, Dr. Prashant. They are like, my very, very good friends, and they are very, very good bioinformaticians also. So I'm like, I always learn from these people. So thank you very much. Thanks to all.
Thank you very much, sir. And we are really honored to have you, have your presence amongst us. And now I would like to request Ms. Pooja to introduce the next speaker.
Dr. Sugnakar is currently working as assistant professor at Lovely Professional University, Punjab. Dr. Sugnakar works on the clinical cancer genetics, cancer epidemiology, and various cancer types. As part of his PhD, he worked on a topic entitled FLT3 mutation screening and SNP analysis of NHEG repair and extrinsic apoptosis pathway in acute leukemia, which involves the screening of low penetrating and oncogenic mutations in the development and progression of disease by studying the genotype associations, based on epidemiological and clinical parameters in the origin of acute leukemia. He also has 7 years of teaching experience for postgraduate and graduate students at Department of Genetics and Biotechnology, Osmania University. He has published 22 papers and 3 book chapters with H&X STEM, trained 50 plus master students. He also is also awarded the Best Poster Presentation Indian Microbiology Association, Best Poster Presentation from IMBIX 2017 Jaipur, a Best Teacher from Joginipalli BR Engineering College, RR District. I request Dr. Sugunakar to take upon the session. Sir, please.
Is I'm audible?
Yeah,
Yes, sir, you are.
audible, and it's a gentle request from team InSyB. Please try to finish in between 15 to 20 minutes. Please try to understand. Yeah, thank you. Thanks for understanding.
Yeah, even I have a meeting at 12:40 at the university, so I'll try to see how can I justify that. Yeah, thank you very much. First, I would like to thank the organizers, especially my guru, my professor, my big mentor, whose support was immense. That is Professor P. Kavkishore. Way back to 2001, 2003, he is my first coordinator for biotechnology when I was doing my MSc. So a big thanks to you, sir, for allowing me to speak something on what work I have done. Thank you so much, sir. And I would like to thank Dr. Jalaja, Dr. Anil, and Rangarao Ambati for giving the opportunity, and also the Vignan institutions. Thank you. Yeah, my topic would today I'll be speaking mostly on envision the role of cancer prognostic biomarkers to intervention of bioinformatics. Okay. Yeah, the presentation would discuss under these segments. One is on clinical front, prognostic biomarkers, ongoing research, which I'm doing. Yeah, I'd love to thank Kenta Nakai yesterday for a briefing about the disease-associated variants in different categories, which was highlighted in BMC Genomics and also in Cancer Translational Medicine, where we are trying to look at, as a part of my discussion, I'll be mostly talking on cancer somatic mutations and with regarding to different diseases, especially with regard to acute leukemia. Yeah. So when of disease-associated variants that we come across is Mendelian disease variant, cancer predisposing germline variants, and recurrent cancer somatic mutations are more serious than that of the complex disease variants. So the background of the study that I've taken is the leukemia. Leukemia is a group of heterological hematological malignancies, which is caused because of the chromosome abnormalities. Presence of immature white blood cells, they're nothing but the blast cells. This is one of the important prognostic marker when you talk about the poor response. And the progression of the disease of acute leukemia is basically depends upon the percentage blast. Absence of mature white blood cells leads to anemia, thrombocytopenia, and leukocytosis. If we can able to see this particular schematic view, we can able to understand that there is a transformation of a normal hematopoietic lineage to an abnormal development that leads to acute leukemia. And the transformation may be because of the dysregulation of cell surveillance mechanism, that is with regard to the genomic instability and also the apoptosis. Okay, the abnormalities include dysregulated DNA repair, failure of cell cycle checkpoints, failure of apoptosis, activation of oncogenes, and failure of tumor suppressant genes. The clinical symptoms like fatigue, shortness of breath, on exertion, easy breathing, pitachi, and other commodities. When we talk about the classification, it all depends upon the rate at which it is progressing, and it is further classified into, the leukemia is further classified into acute and chronic leukemia. I'll be mostly talking about our acute leukemia. It is an abrupt onset of severe disease with immature cell forms of both ALL and AML. When you talk about the hypothesis of the study, when you talk about the hematopoietic stem cells taken into consideration, there's something called as they're lifelong survivors which are prone to damage by various exposures of exogenous as well as endogenous factors. ATM is a sensing gene. They're called as DNA sensors. Okay, when some damage is happening, the first damage is responded by a group of genes, that is ATM and ATR complexes, and this particular ATM will sense the DNA damage and activates the downstream repair pathways. And to the surprise, NHJ pathway is the first pathway to get activated in this hematopoietic lineage. Since it is an error-prone pathway, dysregulated NHJ mechanism might lead to genomic instability and leukemogenesis. The imbalance between the proliferation and the apoptosis associated with the differentiation arrest, it results in survival and clonal selection of the leukemic stem cells, which might contribute to chemoresistance among the patients. The failure of apoptosis and dysregulated error-prone NHJ pathway exert influence in, it can be individually or it can be cumulative impact. Now, as a part of the study, we have collected samples from MNJ Cancer Hospital Institute of Medical Sciences. The total sample size is 772 sample size, and out of which the acute leukemia are 446, which comprises of 225 AML and 221 ALL cases, with a control population of 326 samples. Informed consent was taken from all the subjects, and the inclusion criteria, the primary de novo samples have been taken in the study with a confirmed diagnosis based upon the CBP, bone marrow aspiration, and cytogenetic analysis. Exclusion secondary leukemia and therapeutic was excluded. Ethics committee approval was being taken by both the hospitals, and the epidemiological and the clinical information was obtained from the tumor registry and also with the help of the medical oncologist. Now, the methodology broadly divided into experimental analysis, then we have statistical analysis, then followed by in silico based approaches. I'm skipping this because I don't want to get into in those things. Now, when we talk about the epidemiological and clinical variables, we try and see that when we talk about the age at onset with regarding to the AML and ALL, there is a slight difference between both of the both these disease types. AML basically happens in the elder age group, and ALL happens in the younger age group. You can able to see here clearly that in the group group of 20 to 30 years and more than 30 years, the percentage was 34.2 and 44.9 when compared to the ALL patients less than 10 years, 30.8, and 20 in the age group of 10 to 20 years, 42.1%. And there is an increased elevated risk of male preponderance in developing acute lymphoblastic leukemia in males when compared to the females. It may be because of the exposure. And when we talk about the area of living, the rural population are more prone to the diseases in both AML and ALL, 65.3 and 57.5. It is because of the exposure to the pesticides in the agricultural fields. And with regard to the habitat, we can able to see a double risk of AML patients when compared to the ALL patients, and benzene is one compound which is an important compound in the development of the acute leukemia. And further, we try and talk about the mean SD levels and baseline clinical characteristics at diagnosis. We try to see it that already I've discussed that age at onset, the mean SD at age at onset is 30.88, okay, when compared to the 16.81, and these are the characteristic features that AML is an elder age group when compared to the ALL. And further, the peripheral blast, which I've talked about, the immature blast cells, which are the prerequisite for development of acute leukemia, and the presence of blast cells, which indicate it's a poor prognostic marker, and this shows the poor progression with regard to the disease. And the response rate will be very less when you see high blast percentage. Furthermore, the WBC count, the platelet count, LDH, and disease-free survival have seen a significant increase when compared to the control patients. And now further, how we have the study basically we do it with epidemiological, clinical, and molecular parameters in the development of disease. As a part of the as a part of my study, I've taken NHG repair pathway, including the ATM and also NHG repair pathways, along with extensive apoptotic pathway. We have completely studied the complete set of SNPs which are present in both the pathways, and that's where the imbalance has been seen in with regard to these two. So we have selected a set of SNPs that are present in the FAS receptor, FAS ligand, caspase 8, caspase 3, and caspase 7. And further, to understand its statistical significance, we further underwent to do the interaction studies. A lot of slides have been skipped in the brevity of time. Then we talk about gene-gene interaction studies. There's something called as linkage disequilibrium, multidimensional reduction, combined genotype analysis was being performed. When you have done that linkage disequilibrium analysis, the LD results that what we got from our results indicated that the specific genotypes of NHJ, ATM, and extensive apoptotic pathways may confer risk independently. Because they're present in the different chromosomes, as the alleles are present on different chromosomes, they segregate individually. So that's where we concluded our result that it is both implies to both AML and also ALL. Further, the multi-dimensional reduction analysis has been done, where we have seen the results saying that they always act independently, and it also shown that it has gotten a synergistic interactions between these SNPs of XRCC6, okay, minus 61 C2G and minus 1310 C2G polymorphism may be possibly having an epistatic nature and observed in haplotype analysis. I've skipped that particular haplotype analysis part because of time, where in the presence of two variant alleles conferred protection. When the two variant alleles are present, it is conferred protection, but when each variant allele is present, it is conferring risk. It is conferring significant risk of development. This may be because of the variant allele at one locus may be suppressing the other allele. That's where the concept of epistatic has come into the picture. And the variant allele leading to a normal expression of XRCC6 gene. While the variant SNPs individually might be associated with the NHJ activity. Individually, they're having a risk, but when they're combined, they're showing a protective nature in the normal XRCC6. However, the conclusions have to be confirmed by in silico in vitro studies using cell lines and other studies. Now, this is regarding to the ALL cases. The MRD analysis have revealed the influence of NHJ and apoptotic pathway genes in the origin of ALL. So the synergistic interactions between XRC6 SNPs was evident in both AML and ALL, but the effect of apoptotic genes of ALL was found to be high. The results reveals the lineage-specific variations between AML and ALL susceptibility may be one of the factors. Further, we done combined genotype analysis where we have taken the cumulative adverse genotypes and we have done the analysis and try to see, we've done the odds ratios and try to see how they are reacting. So we have performed an independent analysis of SNPs in both NHJ, ATM, and also the extensive apoptotic pathway. It has shown a significant association of XRCC5 with regard to the VNTR polymorphism with regard to the XRCC5, 6 SNPs, with regard to the FAS genes, and also caspases. Okay, with regard to the one more caspase, it's not only insertion and deletion was taken with regard to the AML. We've seen a significant association between the risk genotypes among the different genotypes in both AML and ALL cases. The same thing was being observed with regarding to the acute lymphoblastic leukemia also. There was a linear increase in odds ratio with respect to the number of risk genotypes in both AML and as well as ALL. There is 5 risk genotypes were taken to taken to the study, which results that a cumulative effect of risk genotypes of NHG and ATM and extensive apoptotic pathway on acute leukemia development. So there is an association of risk genotypes. When they're combined together, the adverse genotypes will facilitate in the development of acute leukemia. It may be possible that dysregulated NHG repair in combination with the low apoptotic ability may predispose to acute leukemia. The genes involved in these pathways are low penetrating genes and may contribute small but cumulative effect if taken together. Now, these are the genotype and phenotype correlation studies that we have performed with regard to the SNP, their location, and the risk allele that is being developed, the genotype, and what is the functional significance. If you can able to see, ATM has got reduced DNA repair capacity, and XRCC has got enhanced DNA repair capacity. These are part of, when you have done our studies, we come to conclusions that there are some significant elevations with regard to each and every thing that is taken into consideration. And when we see the association with regard to the AML and ALL, with regard to the complete remission and the DFS, disease-free survival, there was a reduced expression of this particular genes has been taking place. And before we started doing this analysis, we have done our in silico analysis with regard to the protein-protein interaction studies. It has revealed an interaction between the genes what we have taken. This work was done along with Dr. Prashant at BSIR and Mohan Lata, who are gracious enough to help me in the initial time when we were trying to understand that, when we were trying to develop this hypothesis. This particular image has really helped us to move forward with a very positive note. And further, we have validated the study with regard to the ASPE analysis, with regard to the SNPs that were taken. And further, based upon the classification of the SNPs, based upon their location, we have done different in silico analysis with regard to the SNP location, SNP location in the promoter region, intronic region, and the exonic region. So we have used different softwares. So, for example, as a part of that, we have used a transfect database from the BioBase biological database called as Alibaba 2.1 version. By using a transfect, the presence of a variant allele, you can able to clearly see that. And ATM minus 544, A to T polymorphism. A is converted into T. So there is a variant allele that is coming to picture. The presence of an A allele is giving rise to an SP1, which is giving rise to a series of transcriptional factors binding. But the presence of T allele, you can able to see that there is a new transcriptional factor which has come. Means the variation of a single nucleotide also has an impact on the transcriptional factor binding efficiency. In the inference, says like it revealed the presence of T allele resulted in gain of site of transcriptional factor, which may act as a negative regulator of gene expression, which may act as a negative regulator. Okay. Further, we have studied with regard to the XRCC201 VNTR, then XR6 minus 61 C2G, then 1310 C2G polymorphisms. We are able to see that the presence of this particular, when the variant allele changing, there's a change in the transcriptional factor binding efficiencies, or there's a loss or the gain of transcriptional factors which are highly regulated, and by which these are leading to the disease progression. And further, we combined with regarding to the results by using thermodynamical ensemble predictions. We have done analysis with regard to the XR6 C2G and 1310 C2G polymorphism. We have further done the ssDNA sequence of these two SNPs and the impact of the rate of transcription with regard to the GGG insertion in the promoter region associated with more negative energy may confer stability leading to the optimum expression. This explains the result of haplotype analysis, which revealed that reduced risk of GGG genotype to develop acute leukemia through individually. That's what we try to show in the linkage disequilibrium in the statistical analysis and as well as in the haplotype analysis and also with regard to diplotype analysis. They are conferring risk independently and it has got impact on the epistatic impact is also been present. The list of SNPs that are taken and the factor binding site, we can able to see that these are the different SNPs that are taken into consideration and the presence of this particular associated allele, it is showing different kind of transcriptional factors. So there is always, if you have a for example, you have Fas receptor, if this particular SNP is present, there is a lot of there is a loss of HSC binding site. Okay, there is a gain of site, there is a loss of site, because of which there is an altered expression of gene expression has been noticed. Now further, we have done the intronic analysis, XRCC5 240 G2A polymorphism. We also seen that there is an aberration of the intronic ESS site, which has gotten a potential altering ability. So these are the list of different intronic regions that has been taken as a part of the study, and it has shown that there are some potential splice site has also been created, and it also impact on the on the sequence. Then we further done exonic analysis, and we have seen the energy levels between a normal SNP and also an abnormal normal allele and also an abnormal the variant allele. We could able to see a significant energy levels which has been seen in both the C allele and as well as in the G allele and also the C allele. Exhibited around the free energies were calculated. Okay, and we see that hence the stable mutant RNA may interfere with the wild type during the protein synthesis, thereby resulting in the altered protein levels. So that is what been observed. Now, as a part of the study, we have contributed, we significantly can contribute, and we have recommended that a mandatory screening of FLT3-ITD, which is one of the oncogenic mutation which I have not discussed in the brevity of time. It's a mandatory screening of FLT3-ITD. The presence of this particular FLT3-ITD mutation, the patients will not respond to chemotherapy. So it is when we see in Indian population, in the South Indian population especially, we have seen 18.7% of this particular patients what we have collected in the hospitals, out of which 18% out of for example 226 samples what I have taken, out of which 18.74% have shown FLT3-ITD mutation, and they have to go for different kind of personalized chemotherapy is being given to those particular patients. So presence of FLT3-ITD is very crucial in responding to the chemotherapy. Further, Fas ligand can be an alternative therapeutic target, and ATM XRCC5/6 can be used as biomarkers in the management of acute leukemia. This is first part of study. Then we talked about something called as a prognostic biomarkers. We have different cancer types where we have taken two examples. Have a lot of other stuff. Some of the papers have been highlighted what we have worked on. We have seen in the breast cancer and also in acute leukemia. In breast cancer, we have tried to target this particular mTOR. The PDB ID is 5H64, which is a mammalian target of rapamycin, mTOR kinase, which is an intracellular signaling pathway, which basically helps the cell survival mechanism, the cellular survival, the cellular morphology, and as well as the protein synthesis and integration of metabolism. There are many factors that this particular, many downstream pathway, this particular mTOR plays an important role. The dysregulation of this mTOR pathway, including breast cancer, and it's hyperactivation commonly associated with cell growth and rapid proliferation and neogenesis. So the dysregulation has been caused to that. When you try to see with regard to acute leukemia, as I already talked about the somatic nature of this particular cancer, we try to see isocitrate dehydrogenase, which is one of the important catalyze enzyme, which helps in the oxidative decarboxylation. It mutated version, which will lead to oncometabolite production. That several it disrupts several downstream molecules, which basically undergoes the blockage of the pathway that's taken into consideration. So inhibition of this mTOR and as well as the IDH, mTOR with regard to the breast cancer and also the acute myeloid leukemia. Now we have taken two different, there are important prognostic markers which have been present as a part of the cancer therapy. These are very important. So as a part of this particular study, we have done some in silico based analysis by using the molecular docking and molecular dynamics analysis with regarding to both breast cancer and acute leukemia. We have used different kind of inhibitor molecules which has been taken from the PubChem database. We have used almost a similar kind of protocols for both of the things, and we used a different kind of drug molecules. And this is how we have selected the molecules. The system starting from the suit what have taken by Schrodinger drug discovery suit has been taken, then selection of the protein targets and the known inhibitors were taken into consideration. And we have taken around 40 compounds which selectively consider the best anchored compound, then preparation of the protein, lead compounds and the database preparations, and active site predictions and receptor grid generations have been done. Furthermore, by molecular docking, high throughput virtual screening is being done, and further done with your boiled egg plot analysis. Now, these are the results that we have seen that the compound ZNIC 8556945 showed it has a high affinity with mTOR protein. And further, we have studied the upstream and the downstream analysis with regard to the hydrogen bonding interactions, the electrostatic interactions, then aromatic interactions with regard to which particular amino acids have been targeted and how it is binding. Then we have studied the with regard to the active site with regard to the mTOR protein. And then we have done the boiled egg plot predicting the best 3 compounds to establish drugs by using virtual screening drugs that we have taken into consideration. The lead compound, ZNAC 8556945 is an ideal compound for pharmacophore profiling. The compound shows the best affinity among the pre-exist drugs and inhibited drug against the target mTOR, as well as the study of pharmacophore profiling is also an important criteria. We will encourage, like we encourage to do further analysis with regard to this particular mTOR to prevent the breast cancer, and trying to see how it is going to subside the other pathway analysis. And this is with regard to the IDH of acute myeloid leukemia. The same analysis has been done. We have seen there is one compound called as SHEM BL7 1639 1748 in the PubChem database. We have seen that effective drugs which are basically combining and trying to inhibit the activity of the IDH dysregulated protein. We come across the different binding efficiency studies, and further we got the results. We got very promising results, and we are planning to consider for our downstream work with regard to the treatment in AML in the future. And presently we are undergoing ongoing studies with regard to the erythrosis and prostate cancer, and also the phylogenetic insights into the diversity of human papillomavirus and cervical cancer. Now, this is in collaboration with Dr. Prashant Suravarjala. My PhD student, Dr. Ms. Bhumandeep is working with Prashant and myself. And we're basically looking at the erythrolysis and how it is related with prostate cancer development. We have try to do some correlation studies with the bioinformatics approach. We have enlisted around published, we have taken enlisted the published papers of eukaryotic genes through PubMed gene database, and further analysis is being done. We are trying to see what are the further analysis that we can plan for validation. And as a part of that, we have done some PPA interaction studies where we have tried to see that 22 uric and other 20 associated genes of Homo sapiens using GeneMania and string database was done. It has shown some co-expression of about 51.29 and collocations of 30.59, and then predicted as 8.4 and shared protein domains of 4.79. So the work is still has to undergo, and this is one more phylogenetic insights that we have tried to see with regarding to the HPV cancer with regard to the cervical patients, where we have collected some data from the NCBI. We have retrieved serious sequences from NCBI database, and then we have performed some phylogenetic analysis. Still the work is ongoing, and we have done we try to see what are the high risk and low risk genotypes which have been present as a part of the study. We have done some just a small initial work, and further analysis has to be done. Yeah, I always love this particular quote. The more I study, the more I learn and absorb, the more I realize how truly little I know. But Steve Marabola, thank you. This is from LPU, and I thank the LPU for giving this opportunity and to allow me to do my work. And I would like to thank the big list of people present. My PhD mentor, like Professor S. Vishnupriya, then my medical oncologist, my co-guide, Dr. Raghunath Rao, and the gynecology. Right now, my gynecology, cervical cancer, I'm working with Dr. Leela Digumurthy and Sudha Sinha at MNJ Cancer Hospital, Dr. M. Raghunath, where I did my ICMR postdoc at NIN. He was very kind enough to help me in my analysis. Dr. K. Rajendra Rao from NIN. My collaborators, Prashant, Prash, who is always instrumental enough in helping me in most of the analysis what I'm trying to do. Basically, I'm a wet lab scientist, but when it comes to bioinformatics, I only always look up to Prashant and trying to learn a lot of things from him. And my senior, Dr. Naishara Dunna, who is at Shastra University, Dr. Shailaja, she's scientist at KIMS, Sandhya Amanani, my classmate during my MSc, and Smita Sipawar, the head associate professor, Department of Genetics. My lab mates, Dr. Anuradha, Manjula, Santoshi Rani, Prajita Fani, and Dr. Padala. And above all, what are the work that I have done till today with regard to the clinical cancer genetics? It's all because of the patients who has contributed to do some kind of analysis and trying to help the future prospects where we can really help someone who's really in need. And my PhD scholar Santoshi Kumar Dupala, Bumandeep Kaur, Sadbir, and Pravalika Ala, they're currently working under my guidance. Yeah, thank you.
Thank you, sir. Thank you so much for the insightful talk. And I would like to thank all the speakers who have carried out and delivered their talks in the technical session 3. With this, I conclude this technical session 3 and commence technical session 4. I call upon Dr. S. Asha to introduce the keynote speaker, Dr. Aparna Banerjee.
Ma'am, please. Dear all,
Akila, can you hear me?
yes, sir.
Yeah.
Hello? Could you able
Yeah,
to
madam.
hear
Actually, we
me?
can hear you.
Okay. Shall I start then?
Yes, madam.
Now,
Madam, you're not audible, madam. You're not audible.
We really apologize all the participants for the glitch in the audio. Please cooperate with us for 5 more minutes. We'll fix the problem. Thank you. Hello?
Yes, madam, you're audible. Please start.
Okay. Dr. Aparna Banerjee did her bachelor's and master's in biotechnology, respectively from the University of Burdwan and Vidyasagar University, West Bengal, India. She has obtained her PhD in botany from the University of Burdwan, India, under the supervision of Professor Rajiv Bandopajay. She did her postdoctoral research in Universidad Católica del Mol, Chile, where she is also working presently as assistant professor. In her 6 years of research experience, she has worked on extremophilic microbiology with a focus on bacterial polysaccharides from hot springs. Her research interest also encompasses bioinformatics, genomics, and biodiversity. She has been the recipient of prestigious the National Academy of Sciences India Swarna Jayanti Award in 2017, Indian Science Congress Association Young Scientist Award in 2018. Her present position as assistant professor in Universidad Católica del Mol, Chile, also he is awarded by funds from Government of Chile to work on microbiology of Chilean hot springs. She is also presently alternate director and co-investigator of other funds provided by the Government of Chile. She is also presently Women in Biology co-chair in India's Bioinformatics Society BioQ's organization. She has published more than 25 publications in her relevant fields in nationally and internationally reputed journals with a cumulative impact factor more than 40. I thank the organizing committee for giving an opportunity to read her Dr. Aparna's profile. Thank you. I welcome Dr. Aparna. You can proceed.
Is it visible and I'm audible?
Yes, ma'am.
Okay, thank you. Uh, Thank you to the organizer of International Symposium of Bioinformatics, InSyB 2020, for giving me this opportunity. I'm specifically thankful to Dr. Rangarao and Professor P.B. Kavikishore, sir, both of them, for inviting me in this great event to talk about a part of my research work that I have done. So my today's talk will be on genomic and molecular characterization of a unique avirulent variant of Bacillus anthracis from hot spring. I'm presently working as assistant professor in Universidad Católica del Maule, Chile. And it's like almost 4:15 a.m. in Chile. So I'm trying to do my best, but I also feeling very sleepy to speak at this time. But listening to all the good presentation of everyone today, like I need to give my best. So I will try to give my best presentation. So here is some images of the university where I'm working. And I'm tried to get the images of different seasons because I really like photography. So I tried to take the images from fall season, from summer, from autumn every time. And it really looks very beautiful in different seasons. So now let's start about the talk of today. When we talk about Bacillus anthracis, the bacteria that comes to our mind is Bacillus anthracis. And this Bacillus anthracis, when we talk about this, the incident that comes is the anthrax later attack probably in 2001. And this has been a bacteria that has been used in bioterrorism, and it's a deadly bacteria when we think about anthrax. So there have been some questions that how much prevalent anthrax really is till date, because there have been some debates that anthrax is not that much prevalent now, but it is indeed prevalent. If we see the reports only of last 4-5 years, there are numerous number of cases in different parts of the world. We can see even the map of the anthrax infected countries. A lot of countries around the world, mostly the third world countries, part of third world countries, and the tropical countries are infected by anthrax. So there have been report of anthrax in the wildlife park of Namibia. There have been even report from US about the malhandling of Ebola and anthrax strains. Recently about anthrax, there is one report of how fair it is to use one anthrax bacterial strain or Bacillus anthracis for a COVID-19 vaccine. So that has been now, or even Shigella. So how far it is possible or how much it is free of risk. And there have been also report of anthrax attack in Uganda. So in a lot of countries of Asia and Africa, continuously in last 4 or 5 years, there have been anthrax attack. If we talk about India, the last report that we got is like 10 days back in Assam, and in the national park, they have confirmed that anthrax outbreak happened. And for the reason, it's backdated, but now actually it has increased. So 2 elephants died because of anthrax attack. So we talked about the news report. If we went for the publications related to anthrax, there have been publications in Nature, there have been publication in Nature Review Microbiology that the chimpanzees are also at risk of anthrax. Anthrax is threatening the wildlife continuously. So when we talk about wildlife, do anthrax really infect humans too? Yes, they infect human too. There can be 4 types of anthrax: cutaneous, inhalation, gastrointestinal, and injection anthrax. And anthrax attack is very unlikely. It's not that common, but it's deadly if it happens. So it has already served as a weapon of bioterrorism, as I was talking earlier. Catching anthrax is not like getting a flu or a cold that we get into touch of the other person and we got it. So it's like not that contagious. But the transmission, how that happens? The transmission happens working with the infected animals or with the infected meat products, eating kind of uncooked food that was earlier infected with anthrax from infected animals. So this is the way anthrax got infected from animals to animals. When we see about the genome of Bacillus anthracis, there have been a lot of reports of Bacillus anthracis whole genome around the world, but from India there have been only one report till date, and that one report has been done by our group. So when we also go for the number of whole genome reports per year, we see in 2017 there has been maximum report, and in the reason why this happened and why it started in 2002, because after the anthrax later attack, the whole genome came in 2002 after the first anthrax later attack in 2001. So in 2017, the probable reason can be that in 2015 and 16, there have been a lot of reports of anthrax attacks around the world. So again, in 2018 and 19, it was late, but in 2020, there have been a lot of anthrax whole genome reports around the world, as we can see from the map here. So talking about anthrax, here we are going to talk about a different kind of Bacillus anthracis. And how this Bacillus anthracis is different, because microorganisms that survive in different extreme environment are very different than the other microorganisms. So microbes are basically omnipresent. They are present everywhere around the world in hostile environment, which are otherwise inhospitable to the other group of organisms. They can be present in hot spring, in permafrost, in soda lakes, in nuclear contaminated sites, in deserts, in deep sea, in marine hydrothermal vents, anywhere where otherwise we cannot think of the life. So how the microorganisms survive in that kind of extreme environment? One of the strategy of them is to produce biofilm. And exopolysaccharide production helps a lot in that biofilm production. How they help on that? They permit adhesion. And when the bacteria got adhered to a surface, biofilm formation starts, and then they get protection against the stress factor. The factor can be temperature, salinity, ultraviolet ray, different toxic compounds, even antibiotics, osmotic stress, this kind of things. So basically, we got this Bacillus anthracis from one hot spring of West Bengal. The name is Panifola Hot Spring. And this hot spring is located in a remote coal mine region near a city called Asansol. And the temperature of the hot spring is around 66 degrees centigrade. It varies according to the seasons. And the pH is nearly neutral. But the one important factor in the hot spring was the high arsenic level. We have different work on that. I will not go into the depth of that. But so what we can get from this, that this environment can be called as a polyextremophilic environment. Why? Because the extreme factor was not one, it was more than one. The temperature was high, and also there was abundance of heavy metal arsenic. So we can call this environment as a polyextremophilic environment. So the Bacillus anthracis that we got from there, when we did the whole genome sequencing of the Bacillus anthracis, we found that the Bacillus anthracis was a non-pathogenic thermotolerant variant of Bacillus anthracis. And also from the whole genome, we get some signature sequences of exopolysaccharide production. So we can see how much the genome size was and the GC content. And here is the NCBI accession number of the whole genome too. So some of the important factors that we got of this Bacillus anthracis, the characteristic feature of any Bacillus anthracis is the presence of the plasmids PXO1 and PXO2. Now the uniqueness of the plasmid of Bacillus anthracis is they are very high in size. The PXO1 is around 1.5 MB, and that's a really big size for a plasmid. There have been reports that PXO2 get lost for continuous temperature stress. If we augment the temperature for a long time, PXO2 can get lost, but there have been till now no report of PXO1 getting lost for temperature stress. So this strain was a non-plasmidated strain completely. It was showing similarity with Bacillus anthracis strain Ames and also with Bacillus cereus. As we know that Bacillus, this genus is very complicated, and a lot of time we get very high similarity between anthracis, Bacillus cereus and Bacillus thuringiensis. There have been reports that they are mostly similar. They are one single strain. There were a lot of heat stress related protein that we got over there. EPS-CD operon was present. And another important feature, this was also a non-capsulated strain. the genes encoding the polygamma glutamic acid biosynthesis related, that entire gene, because this basically forms the capsule, that was completely absent in this strain. So it was a non-capsulated variety. There was absence of immune inhibitor A metalloprotease, so there was no previously insect infecting lifestyle in that. There were many additional UV-induced repair capacity related genes present. And another thing that while in the standard Bacillus anthracis strain AMES, there was around 37 prophage genes present, there was only 5 that was in PFAB2. So what is the reason? Because Bacillus anthracis is well known for presence of bacilliphage in this. But what was the reason that there was only 5 phage genes present there? So then we went more deep into comparative genomics and molecular characterization of the strain to get an overall idea. And we make one data set of different Bacillus anthracis strains and also some Bacillus cereus strains to compare this strain. We took the most similar 15 strains with this strain according to the average nucleotide identity. So here we can see the heat map, and in the heat map we prepared 2 separate heat map, one with Bacillus anthracis group and another with Bacillus cereus group. And this Bacillus cereus, this entire group of Bacillus cereus was actually reported from, they were very much alike to Bacillus anthracis, and they were found from an International Space Station report in 2016. So when we see this Bacillus anthracis PFAB2, they were clearly making an outgroup from the other Bacillus cereus. But when we see the Bacillus anthracis PFAB2 from the pathogenic group of the Bacillus anthracis, the top 4, it was also clearly separated from the pathogenic group of Bacillus anthracis. We also did Bray-Curtis similarity index, and Bacillus anthracis PFAB2 was clearly separated from the others. Then we tried to go through pangenomic approach, and what we see that in the we tried to get the pangenome and also the core and accessory genome. The interesting thing that we got through the analysis of prophage in the core genome and the accessory genome, that there was no prophage gene present in the accessory genome. So basically this confirms that there have been no infection of phage in this bacteria earlier, because there was no presence of prophage genes in this, compared to the AMES ancestor, which is the type strain of Bacillus anthracis. So this was one of the unique point, that it didn't have any complete prophage gene present in the accessory genome. This was how it looks, the sub-element classifications. So then we went for the molecular characterization, and it was definitely a strain from hot spring. So it was showing very good growth in high temperature, while the common Bacillus anthracis, their optimum growth temperature is 37 degrees centigrade, and more than 40 degrees centigrade, they just don't grow, they stop the growth, as we can see the strain stern here. But the PFAB2 continued the growth till 55 degrees centigrade. It was also showing the formation of pellicle, more pellicle formation compared to the traditional type strain stern. So here how the pellicle cells look. The PFAB2 cell, Bacillus anthracis, is much smaller in size than the stern, than the pathogenic variety of Bacillus anthracis. While when they form pellicle, it's a characteristic feature of any pellicle formation or biofilm formation that the bacteria, they get elongated in shape because they divide too much and they get elongated in shape during biofilm formation. So another characteristic feature here was that PFAB2 was significantly smaller in size compared to the type strain of Bacillus anthracis. So we also make the spore germination assay, and the what we see interestingly, here we can see the entire transmission electron microscopic study of the spore from the vegetative cell to spore formation to spore germination and the entire spore. So PFAB2 was producing much spore compared to type strain stern. So we wanted to prove that this strain is really a non-pathogenic and an avirulent variety. So we see its association and survival efficiency, association internalization efficiency with type strain stern. And what we have seen, when we infected the Bacillus anthracis with macrophage cells, because Bacillus anthracis grows inside they attack macrophage cells in the body of bovine animals or inside human. So we see that the association and internalization of Bacillus anthracis inside the macrophage cell was significantly less in both the cases, both for association and for internalization, compared to the pathogenic variety of Bacillus anthracis type 10 Stern. We have also seen we have also tried to see in the percentage survival through MTT assay, and the percentage survival of PFAB2 was much more than the control strain Stern. So we also tried to see how the mice model shows the effect, and in mice model, while Stern shows its characteristic death curve, it started showing the death from second day of infection. But PFAB2, in all that continuous 10 days of infection, none of the study mice have died. So basically, through the macrophage infection and MTT assay and in mice model, we have proved that this is really a virulent variety or a non-pathogenic variety of Bacillus anthracis. Now, if we talk in a brief, this strain PFAB2 was producing much more spore compared to the Stern. So we also tried to see the markers present in this. So the three characteristic pathogen marker, toxin marker of Bacillus anthracis, protective and antigen LF and EF, edema factor and lethal factor. These three toxin marker was absent in PFAB2 but was present in Stern. But another marker of membrane marker, FLOT-P, that was present in PFAB2, also present in Stern. So this is a Bacillus anthracis, but this is not producing toxin. And the genetic composition was obviously, it didn't have any plasmid, while the Stern was a plasmid. The Stern is a laboratory strain that was made from Amazon ancestor and is a type strain. But this Bacillus anthracis we got from hot spring. There have been very few reports of Bacillus anthracis actually from extreme environment. One of the report is from Algerian salt lake, and there have been some report from the from air filter of USA. So there have been very less report of Bacillus anthracis from otherwise a non-contaminated area. So interestingly, the pore size of PFAB2, the spore size was very similar to the attenuated Bacillus anthracis strain, which is the name of the strain is Pasteur, and this strain is basically used as a vaccine strain also. So the size of the Pasteur and the PFAB2 is very similar. Also, the length of the spore was very similar to Pasteur. One reason can be when the size is less, they spore much faster. So the lower size probably helped the bacterium to become more potent in becoming pathogenic, but the lack of plasmid in the bacteria again restricted the same. So basically, when we see that this is a non-pathogenic variant of Bacillus anthracis and it is producing big amount of biofilm, so obviously it is producing exopolysaccharide. There have been previously no report of structural characterization of EPS from Bacillus anthracis. So this was the first report of complete structural characterization of Bacillus anthracis. It was showing very high amount of EPS production, around 8 gram per liter of EPS production. It was a low molecular weight EPS, and we see there have been presence of alpha glycosidic bond through FTIR. The temperature, the crystalline transition temperature was also very high, around 276 degrees centigrade. While for basophilic bacteria, the transition temperature is around 260 degrees. For thermophiles, it's always more than that. So from GC and NMR, we have confirmed. From GC, we have seen only glucose is the monomer present in the polysaccharide. And then glucose with alpha glycosidic bond, the option is dextran. And we confirm because beta glucan or levan that have beta glycosidic bond, but because glucose was the only monomer and there was presence of alpha glycosidic bond earlier detected, so we thought this can be a dextran. So from NMR, we have also confirmed from 3 to 6 ppm different anomeric proton resonances, which confirm that Bacillus anthracis PFAB2 is producing dextran kind of polysaccharide. From Bacillus group, Lactobacillus, earlier there have been report of producing dextran. and dextran is very useful all over the world for its application in food industry, in biomedicine as blood substituent, in food industry as a texture enhancing agent. So we tried to confirm that application here too. It was showing more antioxidant property compared to the standard ascorbic acid. It was showing significantly 3.5-fold of that. It was also showing emulsification activity against the vegetative oils. And when we see the rheological property, it was showing very good structural reversibility, a gel-like character, and the phase transition temperature was good. So when we added sodium chloride to the solution of the polysaccharide, it was showing like a thermoresistant gel kind of character. So basically the polysaccharide could be used in food industry as a texture enhancing agent. We also tried to produce nanoparticles using the same polysaccharide because dextran is already reported to be used in green synthesis of nanoparticles by other group of bacteria, by lactobacilli. And we have seen that the Bacillus anthracis PFAB2 mediated polysaccharide, this dextran type of polysaccharide, was producing nanoparticle, silver nanoparticle that was nearly hexagonal in shape. And we can also see the zeta potential was negative, and the average size was within 100 nanometers, so confirming the size of the nanoparticle. And it was interestingly showing very good antimicrobial property against Staphylococcus aureus, against Salmonella typhi, against the fungi Candida albicans, and also Aspergillus niger. So it was showing a broad range of antimicrobial activity against a group of bacteria and fungi. Already we have talked that exopolysaccharides are widely used for their different properties. For here, in case of the dextran produced by this non-pathogenic variety of Bacillus anthracis, it was showing a characteristic like structural material, also emulsification capacity. So it can be used in pharmaceuticals, medicines, or in biomedicines that we have already talked earlier. There can be diverse area of applications. So we can, in a nutshell, we can talk that the heat-loving bacterial EPSs could be an inspiring alternative to food industry as antioxidant or emulsifying agent, or even as biomedicine as alternative of antimicrobial. But all this kind of study, the application of a bacteria was almost impossible if we could not have taken the approach of bioinformatics, the approach of genomics. Because when we talk about a pathogenic bacteria, the story end over there to make an application. So bioinformatics made the path to complete this entire part of the work. And also we got a very interesting answer that we have this idea that Bacillus anthracis is an obligatory pathogen, but science is something new. Every time there is something new to get from the research that we are doing. And here what we have found is Bacillus anthracis, but this is an avirulent variety of Bacillus anthracis. So before I end, I just want to talk about something that I always feel to talk. When we talk about conservation, the entire conservation of microbiology, we only talk about plants, fungi, and animalia. And the big part that is excluded is the prokaryotes, the bacteria and the archaea. They are never included in the mainstream conservation strategy. In 1964, there was the first work of conservation of microorganism, because all these unique bacteria that we are talking about, if we don't conserve them in a time of ever increasing climate change impact and global warming and increasing anthropogenic footprint, we will probably lose their footprint, and even before knowing them. So after that big years gap in 2019, where we talked about symbiotic bacteria and its inclusion, host associated microbiota. But what about all the other microbes that are present in our environment that are not host associated, but they do play an important role in the conservation of our ecosystem in biogeochemical cycle? So there should be a call of microbiome conservation before we lose them. So basically, we should have an integrative conservative management where we will have plant conservation, animal conservation, microbiome conservation all together. And bioinformatics will play a big role there, because bioinformatics is going to save the data set before we are going to lose it. So we need to bridge this gap between computational biology and the field work to get an overall idea of conservation biology. So here I end my talk, and as Prash was saying today morning, it's very important with whom we collaborate and to make our network of collaboration bigger. This entire work was almost impossible without mentioning this name, my PhD supervisor, Professor Rajiv Bandyopadhyay, because the entire work has been done under his supervision and his idea. Dr. Rajiv K. Varshney, without the help of him, this part of the genomics would have been impossible. Professor Rakesh Bhatnagar and Dr. Vikesh K. Somani. We all know that Rakesh Bhatnagar is a very well-known person in the worldwide field of anthrax, and he has been kind enough to allow us to work in the part of the anthrax biology and the molecular characterization of our strain compared to the pathogenic variety of anthrax, because we need to work in BSL-3 when we need to work with Bacillus anthracis, and also to Vasbi Chaudhary and Alex Echeverria Vega for their part of field for the bioinformatics analysis. And I'm really thankful to Bardwan University, to Universidad Católica del Maule, where I'm working, to the department CM, to the project Fondesit, presently what I am doing, and Senbio for the part of laboratory facility. And a big thanks to BioClues organization, because I'm learning a lot of bioinformatics. I was still a step during my PhD, but I am learning every day more with BioClues and the importance of bioinformatics in research every day. So I'm really thankful to all of these people to shaping up my research. Thank you all, and I'm open for some questions.
Okay, move on next. Vignan's Foundation for Science, Technology and Research. I am Akhila, and I couldn't be more pleased to welcome everyone to the valedictory function of this two-day symposium. I extend my greetings to our chief guest, Sri Krishna Devarayulu, Honorable Member of Parliament and also the Vice Chairman of Vignan Group of Institutions. I also welcome the President of the session, Professor K. Ramurthy Naidu, Chancellor of Vignan's Foundation for Science, Technology and Research. Vignan's Foundation for Science, Technology and Research have always been the hub for scientific happenings, may it be projects, publications, technicultural fests or symposia like the happening now. The management is known for always encouraging the students and staff alike to focus on the research and extracurricular activities. The Department of Biotechnology have always been stellar with good academic performance from the students, wonderful teaching and non-teaching staff, emphasis on projects, conferences and symposia. Despite the testing times during the past few months, the department has managed to organize several conferences and symposia and deserves our applause. This two-day international symposium is presented to you under the aegis of AP Bionet, and we hope that these 2 days aided you in bringing new information and enriching the existing domain knowledge. It has been our utmost pleasure to bring forward the symposia to all. Thank you very much from the bottom of my heart. And now I would like to request Dr. Anil Kumar to hand over the session. Sir, could you please unmute yourself?
All right, thank you, Akhila. Okay, greetings to everyone. At the outset, I would like to thank the Vignan management and AP Bionet for helping all the way to make the event a grand successful. The event was organized for 2 days and on the topic entitled International Symposium on Bioinformatics for Disease Therapeutics. It was jointly organized by Vignan University under the aegis of AP Bionet. At this point, I would like to thank Vignan Management for helping in all the ways and Dr. Mohd Asif, AP Bionet President. Thank you, sir. The day one started with the inaugural session presided by our Honorable Chairman, Dr. Lau Rataya, followed by prayer song and the lighting of the lamp. Then the welcome address has been delivered by our Honorable Chairman, following by the release of Book of Abstracts by Chancellor Professor K. Ramurthy Naidu. Later, Dr. M.Y.S. Prasad has delivered the inaugural address, followed by our Registrar Sir, Dr. M.S. Raghunathan. Then Professor Kubanidhi has explained about the Department of Biotechnology, followed by Dr. N. Jalaja about InSyB 2020 and AP Bionet, and Dr. Rangarao for the genesis of AP Bionet. And later the technical session, day one, has started with the keynote delivery by Professor Dr. Kenta Nakai, professor from Institute of Medical Sciences, University of Tokyo. He has delivered on the topic entitled Using Various Kinds of NGS Data for Understanding Cell Type Specific Regulation of Gene Expression, wherein Dr. Kentanaka has delivered on disease-associated variants, chromatic immunoprecipitation, detection of bacterial contamination using sequences, antenna promoter region of Drosophila. Later, another talk by invited speaker, Dr. G. Narahari Shastri, delivered on the topic entitled Understanding the Relationship Between Infection and Immunity, Role of Modeling and Informatics, wherein Dr. Shastri has explained about changing paradigm in drug discovery, personalized medicine, deep technologies for drug discovery. After Shastri, Dr. Bruno Guetta has delivered his topic on bioinformatics to study a range of aspects of immune function, wherein sir has explained about how bioinformatics is useful in understanding immune memory and identification of haplotypes. And before the technical session one was ended with Dr. Ramaswamy, research scientist, Genome Institute of Singapore. Sir has delivered on the topic entitled Career Pathways for Bioinformatics Trained Researchers, wherein he has discussed about data analytics, data literacy, and prediction of algorithms, single cell RNA, and spatial transcriptomics. And the day 2 session has been started with Dr. M, Associate Professor, Perdana University, Malaysia, on the topic Structural Bioinformatics in Drug Discovery, is it still relevant today? Wherein she has discussed about the usage of artificial intelligence in drug discovery, molecular dynamics, docking, and simulation studies. And later we had 6 invited scholars and faculty who has presented their topics. And the day 2 session, it started with keynote address of Professor GPS Raghava, IIIT Delhi, on the topic entitled Computer-Aided Immunotherapy to Fight Against Emerging Pathogens, wherein he has discussed about different types of immunity, confirmation and identification of epitope, and how the epitope can be predicted, and different types of the softwares that their lab has developed, especially prediction of algorithms and different epitopes. Then Dr. Mohammad Asif has delivered a lecture on the topic Mapping the Minimal Sets of Viral Peptidome, wherein he has discussed about antigenic diversity, prediction of epitopes, mapping of the viral peptidomes, and the factors that are affecting the minimal sets. Followed by Dr. Mohammad Asif, Dr. Tikam Chand has delivered on the topic entitled Bioinformatic Analysis of SARS-CoV-2, especially on spike proteins and the steps that are needed in designing the COVID-19 therapeutics, wherein he discussed about transmission of coronavirus, its replication in the host, and immune response of the host towards this SARS-CoV-2, drug discovery, drug repurposing, and importance of this RGD motif. Followed by Dr. Tikam Chand, Dr. Sugnakar Ure from LPU. He has delivered on topic envisioning the role of cancer biomarkers through the intervention of bioinformatics, wherein he clearly distinguished and differentiated the differences between AML and ALL. Simultaneously, he also talked about the effect of pesticides and AML and ALL. In the session 4, Dr. Aparna Banerjee, assistant professor from Chile, she has presented on the topic genomic and molecular characterization of unique avirulent variants of Bacillus anthracis from hot springs, wherein she had discussed about the omnipresence of microbes and how Bacillus anthracis is able to survive that particular temperature or the extreme conditions, and how the biofilms are protecting these things, and later followed by the characterization of the Bacillus anthracis. At the end, I would like to conclude, and once again, I would like to thank Vignan Management from Chairman, Sir, Vice Chairman, Sir, followed by Chancellor, Vice Chancellor, Registrar, Deans, Head Department of Biotechnology, then all the faculty members, followed by my students. Thanks to everyone for making this event a grand success. Now, I would like to say one more thing. Dear all participants, we will send the Google Forms for the delivery of certificates. Kindly fill that one. Once you fill that one, you will get the certificate. And we'll complete this task within 2 days. Thank you for giving me this opportunity. Thank you one and all. Thank you InSyB team 2020. Thank you.
Thank you, sir. Thank you so much for summarizing the symposium. And now I request Dean R&D VFSTR, Professor G. Srinivas Rao, to speak a few words. Sir, please.
Madam, can you hear me?
Yes, sir.
Hello, Professor Naidu is here. Hello, Professor Naidu here.
Yes, sir.
He's not able to hear properly.
Sir, there is a net problem. We are going to fix it soon.
Okay, please. My suggestion is better to move to next. Hello. You better to move to next. I think not able to hear. No.
Yeah.
Dear Akhila.
All right, sir. Please don't mind for the audio glitch. There is a lot of glitch in the internet. Please do cooperate with us. We'll move on to the next speaker. Now, I would like to request Dr. Mohammad Asif Khan to give an overview of InSyB 2020. Sir, please.
Is there any problem? Professor Krishna,
Actually, we are trying to fix it, sir. So, as Mohammed sir hasn't joined yet, so we would like to move to the next speaker. And now I would like to invite Professor K. Ramurthy Naidu, the President of Validity Function and the guest of honor, to take over the session and to give his valuable words. Sir, please.
good afternoon to everybody. Today's chief guest, Honorable Member of Parliament and the Vice Chairman, Vignan Guru.
Let him. Yeah, request him to unmute.
Dean Research and Development, Professor Vijay Ramu, Dean Academics, Dr. A. Ranga Rao, Organizing Secretary of a two-day symposium on bioinformatics, Professor Krupanidhi, Head Department of Biotechnology, Professor Kavi Kishore, Advisor to Department of Biotechnology, distinguished scientists from both abroad and India, who has participated in two-day seminar, symposium. Finally, the faculty members, scholars, and the students. It is my pleasure to give presidential address in a valedictory function of two-day symposium on bioinformatics for disease therapeutics, organized by the Department of Biotechnology. I would like to thank Dr. Ranga Rao and his organizing secretary and his team for organizing the two-day symposium successfully and fruitfully. I appreciate and congratulate to all the distinguished scientists from both abroad and India. This is the platform where the academicians, scholars, scientists, and industrialists are brought in to one place and are benefited much by this two-day symposium. This is the occasion where they exchange their work and innovation shared among the scientists. So it is really needed by not only the biotechnology department, the other departments of our Vijnan University. I am told that the two-day symposium has attracted nearly 1,000 participants from different countries of Asia and the Pacific region of world. I'm so happy for this. I expect that this symposium should organize frequently by the Department of Biotechnology, and I am very happy that the Biotechnology Department has been frequently both the national and international symposium since the 6 years I have been observing. Secondly, experts in the field of bioinformatics have shared their results and knowledge and emphasized the need to develop newer tools and techniques for quickly developing drugs. I'm very happy to note some of the people, scientists, who have given their valuable topics. For example, Dr. Kenta Nikei explained nicely about using various kinds of NGS data for understanding cell type and gene expression. Dr. Narahari Sastry narrated about the relationship between the infection and immunity. Dr. Yam brought out nicely the structural bioinformation in drug discovery. Dr. Fons Fani explained the relationship of biofilm producing glycosyl transferase enzymes, and Dr. Aparna, today's speaker, explained genomic and molecular characterization. Several scientists have articulated nicely the vital role played by bioinformatics on disease diagnosis and disease therapeutics. I'm happy to inform the predictions of human disease using the DNA sequence analysis is a revolution in the medical history. But however, the unlocking of the hidden genetic component for a number of diseases is challengeable nowadays. For example, the cancer, what we have been talking for several decades, it is the proliferation of the cells in the human body. It may be lungs, it may be liver, it may be intestine, it may be throat, or it may be any part of the human body. But in spite of the several efforts, we are unable to discover the medicine for that one, because you know that this particular gene is responsible for the proliferation of cells in the human body. and we are unable to detect the gene component. And I would request the scientists now, you're the real people to find out the way for this remedy for this cancer by inventing the specific gene which is causing for the proliferation of the cells. Yesterday, Professor Krupanidhi said during inaugural session that the more than 5,000 papers have been published regarding COVID-19. And he said further that within 5 or 6 months, the COVID may go out. But to our surprise, 2 days back, a new strain has come out in England and South Africa, Denmark and other countries. So I think we should be very cautious. What happens in future, we don't know. If this COVID-19 goes away, some other disease is coming out. So then I think the scientists always should be very careful, cautious, and think of seriously about even for the new strains. For example, I have been telling teachers are lifelong learners. Similarly, the scientists who have been working in different fields is an unending process. There is no rest for the scientist, and they should be cautious and very aware of all these things. It is a time for think over understand the more infectious nature of the strains that escape the onslaught of medical treatment. The fact is that we are not able to say confidently the vaccine that was said by Pfizer Institute and Bharat Biotechnology, how far it will work out for the existing COVID-19. And we have to wait and see the effectiveness of the vaccine, which the people have been telling that we have come out to the solution for curing, for remedial purposes of the COVID-19. But scientists still, we should be very careful. But I'm very happy that the organization have done very successfully the 2-day symposium yesterday and today. Once again, I congratulate Dr. Rangarao, organizing secretary, Dr. Jalaja, convener, Dr. Anil Kumar, co-convener, the rest of the team and the entire biotechnology department, and for making this symposium, 2-day symposium, more successful and fruitful. Before my closing, I would like to thank once again Dr. Rangarao, organizing secretary, for involving me, for giving an advance information to participate both in the inaugural session and the valedictory session. So I wish once again all of the best of luck, and with the thanks, I remain. Thank you. Hello.
Thank you very much, sir, for your delightful words. And I would like Dr. Muhammad Asif Khan to give an overview of InSyB 2020. Sir, please.
Thank you. Can you hear me?
Sir?
Yeah, can you hear me?
Yes, sir. We can
Okay,
hear you.
thank you. All right, so first of all, I want to congratulate Prof. Kavi Kishore and the organizing committee, everybody involved for a great successful InSyB conclusion to this great successful InSyB 2020. And I think they have done a wonderful job, and they managed to attract a lot of participants. I'm just looking at the number of participants, and the numbers keep going up, and hopefully it'll hit a much higher number than what it is right now. So overall, I think it was very well received. This is probably the highest number of participants that we have ever got for InSyB. So a big congratulations to the committee for that. and maybe among highest. I think last year the Punjab group was also pretty high. Nonetheless, so first of all, congratulations, and a bit of a history of InSyB. So InSyB kind of was born out of the initiative or effort to reach out to countries where INCOB has not gone to. So this is when we were trying to bring INCOB to Indonesia, and we felt that there needs to be more local presence of bioinformatics activities before INCOB could go there, because this is INCOB is more of a conference where the participants are those who have submitted papers. So it was not easy to bring INCOB to all the various Asia Pacific countries. So we felt we needed something else that would be more of an entry-level event that would encourage local participation, especially among young researchers, to be more active and more engaged, and bring people together so that there's a lot of that, so that a critical mass could slowly be built, or if there is already something going on, we could contribute to that ongoing activity. So the idea of InSyB was born through that, and since we were approaching Indonesia to bring INCOB there, it has been many years and INCOB had not been there. So we thought of hosting InSyB first there. So it inaugurated in 2017 in Indonesia with Universitas YRC in Jakarta, and they did a fabulous job for the first InSyB. We received quite a number of participants and speakers from different parts of the world. And then the following year, we went to Malaysia, Selangor, Malaysia, hosted by Perdana University, where I'm based. And then that was in 2018, and the numbers kept going up. And then the following year was in India, Punjab. Hosted by Dr. Harpreet. Very kindly, he offered to help organize this. So this year, we are fortunate to still be in India, and as I mentioned, as was mentioned by Dr. Rangarao, it's on southern part of India. So India is such a huge place, and there's just so much that needs to be done in order to contribute to the ongoing activities in India. There's a lot of excitement. There's a lot of stuff that's happening in India with regards to bioinformatics. So we wanted to plug and play into that equation and do a lot more from our side, and bring exposure through speakers from EpiBioNet. So I think we managed to achieve some of that. We had a good number of contributors from EpiBioNet who spoke at the event today. And so I think in a nutshell, InSyB is slowly achieving its goal, which is it be more localized and connect the local community, and hopefully then this local community would be able to contribute and participate at the global level, hopefully through INCOB or among other platforms out there. And the other focus is, besides local community, is the younger researchers, and I saw a lot of participants here who are our future generation, who will be taking over and doing so much more, and so I think we are also doing a reasonable job there in reaching out to the younger generation. So in a nutshell, I think we're getting there, but more needs to be done. Hopefully one day we'll have actually 1,000 people attending or more into these for these kind of events, to reach out to more future InSyB. We hope that we will be able to attract bids for upcoming InSyB. We don't have a host yet for 2021, but so if anybody is interested, you are most welcome to apply to EpiBioNet to bid for not just 2021, but for future InSyB. Maybe for 2021 we want to go somewhere else and come back to India in the near future. But if you have interest, you don't have to wait for that year. We can start working with you much earlier, much ahead. So with that, I just want to thank Prof. Kavi Kishore and everybody else in the local organizing committee for helping EpiBioNet to complete the year with a successful InSyB 2020. It's been a tight year with INCOB just completing recently, and now InSyB, but I'm really happy to see that it managed to reach to this completion before the end of the year. So thank you again, and I hope to see more of you in future InSyB, whether it's in India or somewhere else. Thank you.
Thank you very much, sir. And I feel honored to have such a great personality, eminent personality like you in front of us. And now I would like to request Dean R&D, Vignan's Foundation for Science, Technology and Research, Professor G. Srinivas Rao, to speak few words. Sir, please. Once again, we apologize for the technical glitch. And I now would like to request the program coordinator in SIP 2020, Professor P.B. Kavakishore, Scientific Advisor, Vignan's Foundation for Science, Technology and Research, to provide his valuable insights of InSyB 2020. Sir, please.
Thank you very much. Thank you very much, the organizers, for giving me an opportunity today to give some of my insights regarding this two-day international symposium on bioinformatics for therapeutic intervention. At the outset, I'd like to thank Dr. Lau Rataya Garu, the chairman of Vignan Group of Institutions, Sri Krishnadevarayalu Garu, Member of Parliament, and also Chancellor Professor Ramurthy Naidu Garu, and Vice Chancellor Professor M.Y.S. Prasad Garu, Dr. Raghunathan, Registrar, Vignan University, Dr. Srinivas Rao, Research and Development, Dean of Research and Development, Dr. Vijay Ramu, Dean Academics, Dr. Krupanaji, Head Department of Biotechnology, for extending all your support, cooperation, and help in many ways in organizing this particular event. I also like to thank, before our Vice Chairman joins this valedictory function, I like to thank not only the management of Vignan University and also the various officials of this university, but all the keynote addressee and also the speakers, invited speakers for the 2 days. I first of all like to thank Professor Kenta Nakai, who has come from Japan and gave a wonderful talk on using how to use various kinds of next generation sequence data for understanding different cell types, specific regulation of gene expressions, and also their utilizations. I also like to thank Professor Narhar Sastry, the Bhatnagar awardee, and also the director, CSR Northeast Institute of Science and Technology, for giving a glimpse of how to understand the relationships between infection and immunity and the role of modeling and informatics. So I also like to thank Dr. Bruno Guetta, Professor, School of Computer Science and Engineering, and also who hails from the University of New South Wales, Australia, for giving a wonderful talk about bioinformatics to study a range of aspects of immune function. Next, I also like to thank Dr. Adikalavan Ramasamy, who is a research scientist at the Genome Institute of Singapore, for giving a very wonderful talk on career pathways for bioinformatics trained researchers. That was followed by a very good talk by Dr. Y. Keat Yam. She has really spoken very well about the structural bioinformatics in drug discovery, is it still relevant or not? She has introduced the artificial intelligence as well as machine learning into bioinformatics. Combinedly, these three tools, that is bioinformatics, artificial intelligence, as well as machine learning, can do wonders. And she has also mentioned that nearly more than 12 drugs have been invented using the tools of bioinformatics. But if you use the bioinformatics along with machine learning and then artificial intelligence, perhaps we'll be able to discover more drugs. That was the essence of her talk. And then Dr. Anil Kumar, who has spoken about some of the known unknowns of the COVID-19. He said the xanthans, how they can bind to the different proteins of the COVID virus, and they make them destructive. Similarly, Dr. Patricio Gonzalez from Chile, he has spoken very well about the computational study on temperature-driven structure-function relationships of the biofilm producing bacteria. And then Dr. Renuka Sorojila has spoken about phytochemical analysis agents as potential targets of the SARS-CoV-2. This is the work basically carried out at Vijnan University, and she is one of the partners here, and she has spoken about that one. And then Dr. Vijay Sai could not participate yesterday because our Vice Chancellor's seminar was there from 3 o'clock onwards. Therefore, we had to skip his talk. And today, Dr. GPS Raghava, former director of IMTech Chandigarh, and who has been working currently at the Institute of Information Technology in Delhi. He has given a wonderful idea of, the predictions of some of the predictions of antigens and other things, how they aided immunotherapy to fight against emerging pathogens. He spoke very well, elegantly. And then, Dr. Prashant Sarvajra, I have enjoyed his talk, listening to him. He spoke very well about the systems genomics of prostate cancer, especially at the Indian scenario or Indian perspective. And then Professor Asif Khan, whose lecture was really enlightening, he spoke about the mapping of the minimal set of the viral peptidomes. And thank you very much, Professor Asif Khan. And then Dr. Tikam Chand Dakal, hailing from Sokhadia University, Rajasthan, he spoke about the role of bioinformatics and how to analyze the SARS-CoV-2 spike proteins using the tools of bioinformatics. And then Dr. Sugunakar Ure from a lovely professional university, he spoke about the cancer biomarkers through the intervention of bioinformatics. And then the technical session was, technical session 4, we had a talk by Dr. Aparna Banerjee, who is hailing from Chile. So all the talks were excellent, and I'm sure that these talks have inspired the students of bioinformatics and biotechnology at the Vijnan University, and also the staff members and scholars of Vijnan University. I'm certain these talks not only just inspired, but they have taken us to a different level of understanding, and then we are certain to say that the talks would be arranged in future at the Department of Biotechnology, Vijnan University, which can help and expand the horizons of the students, scholars, and technical faculty, as well as the faculty members. So I like to thank all the invited speakers. I like to thank all the keynote speakers, and also the short talks given by several people, faculty members. I like to thank them. At the end, I like to say one thing: how bioinformatics has helped, in developing, some of the, and especially in the case of breast cancer, we know pretty well that Herceptin positive, there are two different types of tumors here. or by, which has been unraveled using the tools of bioinformatics. We have Herceptin positive and also Herceptin negative tumors. So we cannot give the same type of medicine for Herceptin positive as well as Herceptin negative patients. So we need to give a different type of medicine for Herceptin positive in comparison with that of Herceptin negative. So in essence, what it means is that bioinformatics tools and techniques are helping us to find out the single nucleotide polymorphisms or mutations that are happening in different, the human beings who are suffering from the same disease. What I mean by that one is, suppose if 100 people are suffering from breast cancer, we cannot give the same type of treatment to all the 100 patients. So this means that we need to first of all analyze the genome of the tumor, and then only a doctor should prescribe the specific and also precise drug. So doctors have to be very, very careful in treating the patients hereafter. So in administering the drugs, they should not administer the same drug to all the 100 different people who are suffering from before administering the drugs. That is one thing. And then it has also, the genome sequence analysis has also helped, for example, in the case of kidney cancer, in the case of lung cancer, in the case of other cancers. In all, there are more than 100 drugs that are now approved by FDA. That is the Food and Drug Administration of America, which have been approved, but developed through the analysis of the bioinformatics or the genomic analysis. So this tells us clearly how important is bioinformatics. Of course, now Dr. Rao Rathe Garu yesterday has emphasized the need to imbibe the principles behind the machine learning as well as artificial intelligence. And we need to combine those tools and techniques to bioinformatics in order to discover the newer drugs that can help us. It's not just cancer that bioinformatics can help, but also today's the coronavirus, which has become a pandemic. So it is important for us to utilize the tools and techniques of all the three, that is bioinformatics, machine learning, as well as artificial intelligence, to combat such pandemics. So thank you very much for giving me an opportunity. I think Vice Chairman sir has just joined, and I hand over the mic to Ms. Akhila.
Thank you, sir. Thank you for the knowledgeable words. And now let me take the pleasure of inviting Dr. Prakash Narayan Reddy to introduce our Honorable Chief Guest, Sri Lao Krishna Deva Railu. Sir, please. Respected Chancellor, respected Chief Guest of the day, Sri Lao Krishna Deva Railu, dear colleagues, invited speakers, and participants of International Symposium on Bioinformatics, InSyB 2020. Good afternoon to you all. I am honored to introduce the Honorable Chief Guest, Sri Lavu Sri Krishnadeva Railu, Member of Parliament to Narsaraopet Lok Sabha Constituency and Vice Chairman, Vignan's Group of Institutions. Sri Lavu Sri Krishnadeva Railu is the son of Visionary Chairman of Vignan's Group, Honorable Sri Lavu Rataya Garu, an eminent scholar, educationist, and socially committed humanitarian. Sri Lavu Sri Krishnadeva Railu has completed his bachelor's in computer science and engineering, followed by media studies and arts at Latrobe University, Australia. He has been serving as Vice Chairman of Vignan's Group of Institutions since December 2008. The Vignan's Group is serving all levels of education from primary to tertiary levels with a current student strength of around 45,000 spread across the two states of Andhra Pradesh and Telangana. During the last 4.5 decades, Vignan's Group is striving towards promoting high standards of education and contributing to socio-economic development of this region. Sri Lavu Sri Krishnadeva Railu is heading, guiding, and shaping Vignan's Group of Institutions by incorporating the best educational practices being followed at premier institutes across the world. Due to his charming personality, humanitarian qualities, Sri Lavu Sri Krishna Devarayulu has contested for first time for Lok Sabha elections and won with a large majority in the parliament elections in India and elected as one of the young parliamentarians in June 2019. He is currently serving as Honorable Member of Standing Committee on Human Resource Development and Member of Consultative Committee on Animal Husbandry, Dairy and Fisheries to Government of India. As a Member of Parliament, he is striving for provision of lift irrigation scheme to irrigate approximately 1 lakh acres of land in the vicinity. And Sir is also striving to provide a water grid, drinking water facility to every household. Apart from that, he is striving to special training to government school teachers and also to uplift the healthcare and establish a medical college. Sir is also striving to develop medium and small scale industries in food processing with primary focus on spices and vegetables. With this brief introduction, I now invite Honorable Sri Lavu Sri Krishna Devarayulu Sir to give his thoughts and remarks on the International Symposium on Bioinformatics InSyB 2020. Thank you for giving me this opportunity.
Sir, sorry to interrupt. We could not hear you.
Sir, we are very sorry to tell that we can't able to hear you.
Hello. Hello. Hello.
Yes, sir.
Yeah, is the problem from here or from there?
Sir, the problem is from Hyderabad, sir, not from Vijnana University.
Hyderabad.
Yeah, right. Sir is in Hyderabad. So there is some internet problem, I think. So we'll fix it, sir.
I spoke from Hyderabad only.
True, but there is some problem there. So we'll try to fix it, sir.
Yeah, okay. Yeah, okay, thank you.
Is it audible now?
Yes, sir. Yes, It is. Please
Okay.
go ahead.
Yeah, thanks for inviting me to speak on the valedictory function of International Symposium on Bioinformatics for Disease Therapeutics. First of all, let me take the names of our Chancellor, Mr. K.R. Naidu Garu, our Vice Chancellor, M.Y.S. Prasad Garu, Professor Srinivas Rao Garu, Dean R&D, Vijay Ramu, Dean Academics, Rangarao Garu, Organizing Secretary for this two-day International Symposium, and Professor Krupalnidhi Garu, Head Department of Biotechnology. And most importantly, the participants who have come across, participated from across the world, Dr. Mohammad Asif Khan, who participated from Turkey, Dr. Kenta Nakai from Japan, G. Narhar Sastry Garu, Professor Bruno Gaeta from Australia, Professor A. Ramaswamy from Singapore, Professor Y. Kayatayam from Malaysia, Professor GPS Raghava Garu from Delhi, Dr. Prashanti from BITS, and Professor Tikam Chand, ML Sukadia, Dr. Sugunakar, Dr. Aparna, and all the other participants who have who I couldn't name them, who have taken up the time and to participate in this today's symposium. Thank you to each one of you. At the outset, I'd like to thank the faculty members of the Department of Biotechnology, Vignan University, for having thought of organizing a 2 days international symposium in collaboration with Asia Pacific Bioinformatics Network. I believe that there are almost 873 participants have registered for this, and they have, with a lot of interest, they have participated in this, and 16 expert scientists, professors have from across the world have shared their knowledge. To start off, I'm not an expert in bioinformatics, but whatever that has been discussed in the last 2 days, I believe that the bioinformatics has potential applications, including drug discovery, personalized medicine, preventive medicine, and gene therapy. Research in such potential areas can take human healthcare to the next level of understanding and helping mankind in getting rid of physical sufferings. Before the Human Genome Project began, researchers knew the genetic basis of only about 60 disorders or so. Today, we know the basis of nearly 5,000 conditions. This is purely because of the use of bioinformatics tools and techniques. Prescriptions are also changing because of human genome sequencing and medical genomics. More than 100 different Food and Drug Administration approved drugs are now packaged with genomic information or DNA information that tells the doctor to test their patients for genetic variants linked to the efficiency, dosage, or risky side effects. Taking cognizance of the facts mentioned, students, scholars, faculty members, and scientists alike can get inspired to take initiatives for expanding their research horizons that bring laurels, especially to the Vignan University and also to the whole of India. This bioinformatics is the future of medical care and drug care. So I once again congratulate the faculty members of bioinformatics and biotechnology for their relentless endeavor to invite speakers from other countries and make this international event a success. I also hope that 2-day deliberations have successfully and extremely useful to the entire academia and scientists, and more importantly, the participants. I hope to see in the next few months to years more number of doctorates. These participants will pursue doctoral studies in these fields and also publish a good number of papers that will be useful for the not only for India but across the world. Finally, I thank the organizers for giving me this opportunity, and once again I congratulate the organizers for putting all this effort in bringing so many people together and in making it a grand success. Thank you once again for inviting me.
Thank you very much, sir. Thank you for your valuable words. And I now would like to request Dr. N. Jalaja, Department of Biotechnology, Vignan's Foundation for Science, Technology and Research, to give feedback report from students of Vignan's University. Ma'am, please.
So, greetings to everyone. In respected Chancellor Sir, Vice Chairman, and students, deans, everyone. So now I would like to give a feedback on the Vignan Foundation students on International Symposium. InSyB 2020, organized by Department of Biotechnology, Vignan University in association with AP Bionet, is informative and insight to all the students. A symposium promoted bioinformatics as an intense tool for the understanding of biological information, be it a virus, bacterium, plant, or human, and also brought awareness on all younger generations. Students understood bioinformatics is an interdisciplinary project, helps to develop methods and software tools for understanding biological data better, especially when the data sets are huge and highly complex. Biotechnology students and the scholars got aware that along with the laboratories have to handle data, otherwise it will become extremely difficult, and also their analysis without the computer tools and techniques. Indeed, bioinformatics combines biology, pharmacy, computer science, information engineering, mathematics, and statistics to analyze and interpret the biological data, especially the DNA sequence analysis. Students' conception is bioinformatic tools derive some meaningful information out of the human genome sequences and also the career in the human genome projects. Students felt it is a unique opportunity to listen to experts in the field, which can help to extend their horizons. The proceedings motivated the students and scholars alike and helped to shape the destiny in the field of disease, diagnostics, and therapeutic interventions. Students also interpreted that more than 100 drugs developed through DNA data analysis have been approved by Food and Drug Administration and are being used for therapeutic interventions. Students sense that time is ripe to unfold the secrets behind the life's process and also the disease progenesis in humans and animals. This is possible only when we have powerful computer software tools and programs and specific analysis pipelines, especially in the field of genomics. Students requested the faculty to conduct more and more symposiums at the Department of Biotechnology, Vignan University in future to shape their career in research and to get the industrial awareness for their development and growth. So thank you for giving this opportunity.
Thank you, ma'am. Now I would like to call upon Professor T.C. Venkateshwarlu, Department of Biotechnology, Vignan's Foundation for Science, Technology and Research, to deliver his word of thanks for this session. Sir, please.
Yeah. So very good afternoon to one and all. So today's international symposium went on well. So now we're closing the session with the vote of thanks. So I deem it a great honor and privilege to propose the vote of thanks on the occasion of the valedictory function of this international symposium on bioinformatics for disease therapeutics, organized by the Department of Biotechnology, Vignan's Foundation for Science, Technology and Research under the umbrella of Asia Pacific Bioinformatics Network. So firstly, I would like to extend my deep sense of gratitude to the office bearer of the Asia Pacific Bioinformatics Network, especially Professor Asif Khan, for providing an opportunity to bring the researchers, pharmacists, academicians, and also industry experts onto a single platform so that can help the students, scholars, and faculty members to further their knowledge. Interactions with such experts in the field certainly enhances the academic excellence and also accelerate the research in the field of bioinformatics. I would like to express my heartfelt thanks to the keynote speakers of this international symposium, Professor Kenta Nakai and Professor GPS Raghawa, for enlightening us with their research results. I want to extend a special thanks to the all invited speakers, namely Dr. G. Narasimha Narahari Shastri, Shanti Swarup Bhatnakar, Professor Guno Gita, Dr. Aday Kalavan Ramaswamy, Dr. Vaikit Yam, Mr. Patricio Fane, Dr. Renuka, and colleagues, Dr. Abhinav Parashar and Dr. Anil Kumar, for sharing their valuable knowledge and experiences with the participants. So this international symposium gave deep insights and also some interesting facts on disease therapeutics. So the interesting topics such as tissue-specific expression of genes using NGS tools, computational biology in infection and immunity, sequencing rearranged immunoglobulin genes, career paths for bioinformaticians, structural bioinformatics, and big data analysis, etc. are covered in this symposium. I express special thanks to Professor S. Krupanidhi, Head Department of Biotechnology, for valuable guidance and encouragement in all aspects in organizing the symposium. I should not forget to thank the convener, Dr. N. Jalaja, and the organizing secretary, Dr. A. Rangarao, and co-convener of this symposium, Dr. S. Anil Kumar, who worked tremendously for making this symposium a successful one. I am thankful to our university administrators, namely our beloved chairman, Sir Dr. Lau Rattaya Garu, Vice Chairman Sri Krishna Devarayal Garu, Chancellor, Vice Chancellor, and Registrar for sharing their valuable time with our participants and students in the symposium. Without the strength of the participants, this symposium would not be successful. I appreciate all the participants for attending the sessions and listening to the lectures. I also appreciate the help, support, cooperation rendered by both teaching and non-teaching staff of the Department of Biotechnology. So especially, I thank the chair and co-chair of this symposium, the faculty of Biotechnology Department, namely the Jagdish Kumar, Abhinav Parashar, Srikant, Anjani Devi, John Babu, Najinin Bobby, Asha, and Venkatanarayana has served as session chairs for this symposium. And the student voluntaries, Pujasri, Akila, Padmini, Suhana, Vaishnavi, Dheeraj, Ajay, Raju, and Dwaraka, and other students have helped a lot for success of this international symposium. So last but certainly not the least, I would like to express my utter sincerity to the Vignan governance for appreciating our efforts and providing us this opportunity. Thank you all.
Thank you very much, sir. And now I would like to call Bhavya Shri and Lasya for national anthem. All of you, please stand up. Thank you.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्छल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मागे गाहे तव जय गाथा जन गण मंगल नायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे.
Thank you very much, Lasya and Bhavyashri. And I would like to inform all the participants that we'll share the feedback link shortly. After filling the feedback link, certificates will be provided through your mail. We are sure that it may take for maximum of 2 days for delivering the certificate. If any queries, please contact the given addresses and the given phone numbers in the chat box. Thank you. Being a part of organizing committee of InSyB 2020, we thank each and everyone who has participated in this session. Thank you very much.
Thank you, Dr. Akila and Pujita, for being anchoring the sessions for last since 2 days. Yeah, thank you very much. Now I'm closing the session. Thank you one and all for participating. Thank you. Bye.